Good afternoon, all. My name is Bernie Rossoff, and as chair of this study, I would like to welcome everyone to the third public meeting for the assessment of NIH research on autoimmune diseases. This is, as most of you know, a congressionally mandated study that is sponsored by the National Institutes of Health. I am joined by the committee members who are all participating virtually. I will ask them in a few moments to introduce themselves. I want to note that this is an open, on-the-record session. Interested individuals have been invited to attend as observers via web conference. And I'd also like to remind everyone that this is an information gathering session. Important for us to recognize that is, the committee is in the process of assembling materials that it will examine and discuss in the course of making its findings, its conclusions, and recommendations. Therefore, I ask everyone here today to understand that this time will be for gathering information. And everybody should recognize that the committee has made no conclusions. In addition, comments by individuals, including invited speakers and members of the committee, should not be interpreted as positions of the committee or of the academies. Committee members typically ask usually some probing questions in these information gathering sessions that may not be indicative of their personal views, but really for the purpose of being better informed. So for all of us on the committee, and even those in the audience, this should be information gathering, and I am certain that we will be appreciative of the information that we will gain from today's meetings. The committee will deliberate thoroughly before writing its draft report. Moreover, once the draft report is written, it must go through a very rigorous review by experts who are anonymous to the committee. And the committee must then respond to this review with appropriate revisions that adequately satisfy the Academy's report review committee and the National Academy's president before it is considered an official Academy's report. The public sessions will be held today and tomorrow. Today's focus will be to hear a panel of representatives from institutes that fund intramural and extramural research on autoimmune diseases at the NIH. Tomorrow, we will hear from experts speaking on specific autoimmune diseases. We will start off with presentations from representatives from 4 NIH institutes that fund autoimmune work: the National Human Genome Research Institute, the National Institute of Allergy and Infectious Disease, the National Institute of Environmental Health Sciences, and the National Institute of Arthritis and Skin Disorders. Dr. Daniel Kastner, Dr. Carol Barron, Dr. Lisa Ryder, and Dr. John O'Shea will discuss the intramural research for autoimmune diseases at their respective ICSs, including recent scientific advances from funded work, any evolution or change in funding priorities, identification of possible research gaps, and their approach for setting priorities for funded work. After a break in a while, we will continue the public sessions by then focusing on the extramural side of NIH research. And we will hear from representatives from 3 NIH institutes: the National Institute of Diabetes and Digestive and Kidney Disease, the National Institute of Environmental Health Sciences, and the National Institutes on Minority Health and Health Disparities. Dr. Lisa Spain, Dr. Michael Humble, and Dr. Raja Pakshu will discuss the extramural research for autoimmune diseases at their institutes. And they too will be addressing recent scientific advances from funded work, evolution in funding priorities, and possible research gaps, and their approach for setting priorities for funded work. Regarding the format for today's talks, after each speaker's remarks, the committee will be able to ask brief qualifying questions. Then, after all presentations have been delivered, there will be a 35-minute discussion. All attendees can indicate their questions or comments at any time during this meeting time by typing them into the Q&A box at the bottom of your screen. The Academy's study team and myself will be tracking comments for speakers and the committee. Those who would subsequent to this meeting, like to talk with members of the committee are kindly asked to touch base with the staff officer, Henrietta Awo Osai-Anto, by emailing autoimmunestudy@nas.edu. That is autoimmunestudy@nas.edu. Once again, this meeting is being recorded. Now, I'd like to ask the committee members to introduce themselves, giving their names and current affiliations, recognizing that brief bios of all committee members are available online. My name is Bernie Rossoff. I'm a gastroenterologist, professor of medicine at the Zucker School of Medicine at Hofstra Northwell, board member at the Northwell Health System, and chair of this committee. Glinda? I think I saw Glinda before, but if not.
Committee members, please unmute and unmute yourselves and put on your videos.
Well, I'll just move down at the moment to Deirdre and come back to Glinda.
Sure. I'm Deirdre Cruz. I'm a nephrologist and professor of medicine at Johns Hopkins, and also associate director for research development at the Center for Health Equity.
Bill?
Yeah, I'm Bill Duncan. I'm an immunologist and a vice provost of research at East Tennessee State University with a lot of experience in managing large international trials and national trials in infectious disease, autoimmune disease, and organ transplantation.
Delisa?
Yes, I'm Director of Translational Research in the Cardiovascular Department at Mayo Clinic with a long time history in studying viral and environmental myocarditis, and also interest in sex and age differences in autoimmune diseases.
Sonia?
I'm an associate professor of medicine at Harvard Medical School, and I work at Brigham and Women's Hospital. And I'm a gastroenterologist specializing in Crohn's disease and ulcerative colitis. And my research has been on reproductive issues in IBD.
Lisa Iazzoni, one of the three Lisas here today.
Thank you, Bernie, for distinguishing me that way. My name is Lisa Iazzoni. I'm professor of medicine at Harvard Medical School based at the Massachusetts General Hospital. I'm a health services researcher, and my focus is on healthcare disparities for people with disability.
Andrea?
So I'm an assistant professor at the University of Toronto and a pediatric rheumatologist at the Hospital for Sick Children. I have a clinical research interest in lupus, particularly neuropsychiatric lupus, as well as mental health for young people with rheumatologic disease.
Scott?
Hello, I'm Scott Lieberman. I am a pediatric rheumatologist and associate professor of pediatrics at the University of Iowa Carver College of Medicine. And my research lab focuses on understanding the early immunological events in the development of Sjogren's disease.
Michael?
I'm a professor of medicine and obstetrics gynecology at the Hospital for Special Surgery, Weill Cornell. And for 7 years, I was extramural director, then acting director of NIAMS.
Jill?
Hi, I'm professor and chair of epidemiology at the Colorado School of Public Health. My research has focused on the nutritional and genetic risk factors for autoimmunity and subsequent autoimmune disease.
Emily?
Hi, I'm Emily Summers. I'm an associate professor and epidemiologist at the University of Michigan. My primary appointment's in rheumatology. I focus mainly on autoimmune diseases, including lupus, and I have a strong interest in environmental health as well as women's health in relationship to the autoimmune and musculoskeletal disorders.
Barbara?
Barbara Vickery. I'm a chair of neurology at the Icahn School of Medicine at Mount Sinai. I'm a health services researcher with interest in work in developing interventions to improve quality of care for neurologic conditions.
Glinda?
Hi, I'm late. I'm Glinda Cooper. I'm an epidemiologist with experience mostly with the systemic autoimmune diseases.
Well, let's introduce the National Academy team. By the way, without them, this is all not possible. So I always feel comfortable introducing them and congratulating them for all the work that they do. Awo?
Yes, good afternoon, everyone. My name is Awo Sayanto, and I'm the study director for this study. And there's Kristen.
Yes, hello. My name is Kristen White, and I'm an associate program officer on this study.
Dara?
Hi, I'm Dara Rosenberg. I'm the research associate for this study.
And Layla?
Hi, everyone. I'm Layla Maiden, and I'm the senior program assistant for the study.
Thank you. And the board director for, under which the study sits, Rose Martinez, is also on this line.
Good afternoon. I'm Rose Martinez. I direct the board on population health and public health practice.
So once again, thank everybody for being here on this afternoon, and I'm looking forward to the next 4 hours of discussions and information gathering. So we'll begin the NIH intramural portion of our panel discussion today, and I'll introduce the 4 speakers. The speakers will have approximately 20 minutes, and we'll follow those 20 minutes by 5 minutes of clarifying questions that the committee may have related to the content of the presentations, which may be helpful to the committee for their work. Let me quickly go through the speaker bios. By the way, also available. Daniel Kastner is the scientific director of the Intramural Research Program at the National Human Genome Research Institute. He is a distinguished investigator in the metabolic, cardiovascular, and inflammatory disease genomics branch. Previously, he was at the National Institutes of Arthritis and Musculoskeletal and Skin Diseases. He and his group moved to NHGRI in 2010. The inflammatory disease section identifies genes underlying inherited human disorders of inflammation, elucidates their function, and applies these insights to the diagnosis and treatment of disease. Carol Barron is a pediatric rheumatologist, and she is the deputy director of the Division of Intramural Research at the National Institutes of Allergy and Infectious Diseases. Her research focuses on autoinflammatory disorders. Lisa Ryder is the acting head and senior research physician of the Environmental Autoimmunity Group in the Clinical Research Branch of the National Institutes of Environmental Health Sciences. The group conducts a broad program of clinical, translational, and basic investigations in the areas of adult and pediatric autoimmune diseases. Dr. Ryder has focused much of her work on autoimmune muscle diseases in children. John O'Shea is the scientific director and director of the Intramural Research Program at the National Institute of Arthritis and Musculoskeletal and Skin Diseases. Previously, he served as the chief of the lymphocytic cell biology section of the Arthritis and Rheumatism branch at NIAMS, and also as the chief of the Molecular Immunology and Inflammation branch. Dr. O'Shea moved to NIAMS from the National Cancer Institute, where he had started his own group. Dr. O'Shea has also served as acting director of the then named NIH Center for Regenerative Medicine, dedicated to the stem cell science. So let me begin with Dr. Kastner. Daniel?
All right, let me just share my screen here. And we'll get the presentation up. Okay, well, thank you so much, Dr. Rossoff, for that kind introduction. And as Dr. Rossoff said, I'm the scientific director of the Intramural Research Program of the National Human Genome Research Institute. But in fact, the work that I'm going to talk to you about actually has been incubating, so to speak, for the last more than 3 decades, starting when the National Institute of Arthritis and Musculoskeletal Diseases was in its infancy in the late 1980s. And in fact, the work that I'm going to tell you about has been a very, very wonderful collaboration between the National Human Genome Research Institute for about the last 10 years, as well as the NIAMS, and also the National Institute of Allergy and Infectious Diseases. The title of my talk is Horror Autoinflammaticus: Autoimmune Disease Research in the NHGRI Intramural Research Program. And this title actually harkens back to the concept of horror autotoxicus that was put forth by the great immunologist of the beginning of the 20th century, Paul Ehrlich, who was the discoverer of antitoxins and had proposed the idea that, in fact, the immune system is so potent that autoimmunity would be impossible, that, in fact, any sort of immune attack on the host would be so devastating that it would be impossible to happen. Well, of course, we know over the course of the 20th century, the mid-20th century, that in fact, immunologic responses involving the adaptive immune system, that is to say, antibodies and antigen-specific T cells, do mediate both organ-specific and systemic autoimmune diseases. And towards the end of the 20th century, it became clear that, in fact, innate immune activity that might be misregulated can also cause unprovoked inflammatory manifestations. And that's what we'll be talking about in the course of this talk. Before we go into that, though, perhaps I should just give you a little bit, for those of you who aren't familiar, of the essentials of the NIH Intramural Research Program writ large. So this is just having to do with any intramural program at the NIH. And I've just come up with a few salient features of intramural research at the NIH. First of all, of course, the budgets are appropriated by Congress and administered through the institutes and centers. That is to say, investigators get their money through this stream rather than by applying for grants, as would be the case in the extramural community. Secondly, although this is not written in stone, in fact, in most of the institutes, there is an institutional commitment to researchers over projects, sort of like the Howard Hughes Institute, that in fact, the commitment is to specific investigators rather than that there's the commitment to a particular project. Thirdly, the review of intramural research occurs usually on a quadrennial basis, and oftentimes it is heavily retrospective, whereas extramural research is oftentimes reviewed in a prospective way. In the intramural program, the review is more retrospective by outside reviewers, the boards of scientific counselors. Fourth, the intramural program, because of relative stability in funding, oftentimes encourages long-term studies that wouldn't be possible in other settings. Fifth, because of the fact that there is the retrospective review and the long-term commitment, there's the opportunity for high-risk, high-reward projects that would be difficult to do with R01 funding. And in fact, in the NHGRI, we certainly encourage our investigators to take on high-risk, high-reward projects. And then finally, there are specialized resources that are available to intramural investigators. That is to say, things like the Clinical Center of the NIH, the largest hospital in the world that is dedicated solely to clinical research, and where there is no cost, either to the investigator or to the patient, to be studied at the clinical center. So these are, of course, extraordinary opportunities. And in the intramural program of NHGRI, and I would say that it would be fair to say this for all of the institutes of the NIH, that the apportionment of resources is very, very attuned to the advice that we get from our boards of scientific counselors. So we have a rigorous review process, and we do pay attention to the advice that we get. With regard to the NHGRI itself, it's the Genome Institute. And so we focus on basic genomics. We focus on the development of new technologies and bioinformatics. We focus on at least some inherited diseases, and fulfilling the ELSI mission of the NHGRI. We also have a component of the intramural program that studies social and behavioral research. Altogether, we have 24 tenured senior investigators, three tenure track investigators, 3 senior scientists, 2 senior clinicians, a number of associate investigators, which is like a research faculty position at a university, 9 adjunct investigators, and 1 scientist emeritus. Altogether, we have a staff of around 625 individuals, including these faculty members, and our overall appropriated budget is around $120 million. We are spread out in 7 buildings on the NIH campus, as well as having facilities in Rockville and Baltimore. So in any case, we have a very distinguished faculty. 10 members of our intramural program actually are members of either the National Academy of Sciences or the National Academy of Medicine, or both. And I'll just show you photos of them. The first is Diana Bianchi, who was elected because of her work on cell-free DNA in the circulation of pregnant women and the diagnostic potential of that. She is, of course, also the director of the National Institute of Child Health and Human Development. Les Biesecker, who was elected because of his work on somatic mosaicism and Proteus syndrome. Francis Collins, who was elected because of his pioneering work in the Genome Project early on. Josh Denny, a relatively recently elected person, compared with Francis anyway, who was elected because of his work on phenome-wide association studies, and he is the head of the All of Us initiative at the NIH. Bill Gall, who was the initiator of the Undiagnosed Diseases Program in the United States, and also the Undiagnosed Diseases Network worldwide. Gary Gibbons, who was elected because of his work on health disparities research, and he is, of course, also the director of the National Heart, Blood, and Lung Institute. Myself, I was elected because of my work on autoinflammatory diseases. Elaine Ostrander, who was elected because of her work on dog genetics, canine genetics. Charles Rotimi, who was elected because of his work on African diaspora populations and their genomics, and Julie Segre, who was just recently elected because of her work on the skin microbiome. So we have a very established and distinguished faculty, and as you might imagine, in terms of steering them to do something other than what they have been doing in their careers, that would be a tall order, although we can encourage them to take on various autoimmune diseases. We also have a new member of our faculty, Dr. Lindsay Criswell, who's actually the new director of the NIAMS. And institute directors have to have their research program in another institute. And since Dr. Criswell does a lot of work on the genetics of autoimmune diseases, she has joined the NHGRI Intramural Research Program, and we expect that we will be augmenting our work on the autoimmune diseases. She works on Sjogren's syndrome in the coming year or two as she establishes her program. We also have a new tenure track investigator, Neil Hanchard, who was a Rhodes Scholar from Jamaica 20 years ago and now has joined our institute studying complex childhood diseases, genetically complex childhood diseases. And then another of our young stars, Adam Phillippe, whose grandfather actually was an engineer in Hershey, Pennsylvania, who developed a machine that made none other than the Hershey's Kiss. But his distinction, Adam's distinction, is that he's a tremendous bioinformatician who's an expert on analyzing sequence data from the newest generation of sequencers that sequence very long single molecule reads. And this has allowed basically the assembly of a new human genome, basically, with no gaps in it. Whereas the genome that was assembled back 20 years ago had a number of gaps in it. This new human genome, in fact, doesn't have gaps in it. Adam just published a paper in Nature on a gapless sequence of the X chromosome, and we expect that the complete sequence of the human genome, which is roughly 5 or 6% more sequence than what we had before, will be coming out hopefully either later this year or early next year. So in any case, to talk a little bit about the autoinflammatory diseases, I already mentioned that the adaptive immune system and the innate immune system are different. And in fact, we know that the adaptive immune system involves T and B lymphocytes as the major players. The receptors somatically rearrange and mutate, and autoimmune diseases are characterized by high titer autoantibodies or antigen-specific T cells. The innate immune system, on the other hand, has myeloid lineage cells as the major effectors. The receptors are hardwired in the human genome, and the autoinflammatory diseases are characterized by seemingly unprovoked inflammation without the high titer autoantibodies or antigen-specific T cells. I started work on what turned out to be the autoinflammatory diseases back in the late 1980s as a fellow in the new NIAMS. And I was interested in using the tools of the Human Genome Project to solve questions with regard to the pathophysiology of various unexplained disorders of inflammation, some of them characterized by unexplained arthritis. So in any case, I, over the course of several years, established a project to find the gene for familial Mediterranean fever, an unexplained condition, at least at that time, by the new process of positional cloning. Altogether, it took from 1989 to 1997, 8 years to find the gene. And this is just an example of this kind of long-term, high-risk, high-reward project that is the hallmark of intramural research, or at least it can be the hallmark of intramural research. When we found the gene, it was an example of be careful what you ask for because you just may get it. We found a gene that was totally novel, but the good thing is that eventually we learned that it is the prototype for a whole family of genes that are involved in regulating the innate immune system. And there is a domain at the N-terminus of the protein, the encoded protein, that is now referred to as the pyrin domain that is found in some 20 different proteins that are involved in the regulation of innate immunity. Subsequent to that, we went on to discover another gene that causes a periodic fever syndrome, the P55 Tnf receptor gene. And that mutations in that cause a disease called TRAPS. We published that in 1999. And from that, we proposed the concept of autoinflammatory disease. This was a new idea, at least at that time, 20 years ago. Subsequent to that, we recognized, and others recognized, that there's in fact a continuum running between autoimmune diseases at one end of the spectrum and autoinflammatory diseases on the other end of the spectrum. That was proposed in this article that's up in the center of the screen. And then subsequent to that, we and others learned about molecular mechanisms of these diseases. And the field of autoinflammation has now grown substantially, where in fact, the last meeting of the International Society of Autoinflammatory Diseases before the pandemic was in Genoa, and almost 700 people attended that meeting. And there is now a textbook of autoinflammatory diseases, some 800 pages long, and the number of autoinflammatory diseases is somewhere around 40, over 40. Another of the important aspects of the autoinflammatory diseases is that they represent mutations in molecular lesions in various molecules that are very important in regulating the innate immune system. And that has allowed us to predict that certain kinds of targeted therapies would work in these patients. And so one of the diseases that we've worked on is a disease called Nomid. neonatal onset multisystem inflammatory disease. It's caused by mutations in NLRP3, which is an important molecule that regulates the inflammasome, a macromolecular complex that's involved in IL-1 activation. And you can see on the top of this slide some of the clinical manifestations of untreated Nomid. Patients have an urticarial skin rash. They can have conjunctivitis. They can have, as shown in the MRI of the brain, chronic aseptic meningitis, the white representing inflammation of the meninges. And sometimes they can have inflammation of the cochlea, the arrow pointing to the inflamed cochlea on a FLAIR image of the brain. Treating these patients with an IL-1 inhibitor has dramatic results in terms of the clinical picture of these patients. You can see that within days, the urticarial rash goes away, the conjunctivitis goes away. Within 3 months, the meningitis is largely resolved, and the cochleitis is resolved as well. So this is extremely gratifying for the patients who otherwise, I can tell you that the natural history of this disease would be severe intellectual disability or death by young adulthood. And we're now seeing patients that we treated early with IL-1 inhibitors back in the early 2000s who are now college students, at least one of them majoring in biochemistry. So it's a very gratifying thing, but scientifically, it's also extremely gratifying because one would have made the prediction based on the disease, based on the clinical phenotypes, based on the role of NLRP3, which was becoming evident from basic science studies, that an IL-1 inhibitor would work in these patients. And there are a number of other diseases that we don't have time actually to talk about in detail. This is another one relevant to autoimmune diseases, a disorder that we call DADA2, deficiency of ADA2. It was discovered by next-gen sequencing back in 2014 or published back then. And it's a disorder in which children present with recurrent fevers and strokes, but it also is a cause of polyarteritis nodosa. And we now know from studies that our group has done and published in the New England Journal in 2019 that TNF inhibitors are extremely effective in preventing both the strokes in these patients as well as the PAN manifestations. In the last year, we've published several articles that have advanced this field. This is a paper in Nature that we published on a new disease called CREA syndrome, in which patients have mutations in RIP1, RIP kinase. The mutations render RIP kinase uncleavable by caspase 8, which is usually the enzyme that degrades it and turns it off. And this leads to an inflammatory syndrome in which patients have fevers, lymphadenopathy, and organomegaly, and many of the patients respond to IL-6 inhibition. We've also studied Behçet's disease over the course of the last several years, have identified some genes that are genes that have common variants that are associated with Behçet's susceptibility. And in fact, we found a case of a single gene that acts as a high penetrance monogenic cause of Behçet's disease, TNF-AIP3 up in the upper left-hand corner. But in any case, the interesting thing about this is that several of these genes that have common variants associated with Behçet's susceptibility also line up with genes that were discovered by the direct-to-consumer company 23andMe as being genes, variants of which are associated with canker sores, common canker sores. And actually, these same genes are circled here on this GWAS of canker sores that are also susceptibility loci for Behçet's disease. That led us then to think about FAPA syndrome, which is the most common periodic fever syndrome in children, where again, variants in these genes lead to susceptibility to FAPA syndrome. In fact, when one looks at HLA associations, one can see a difference between Behçet's disease, where there's an association of a class 1 molecule, B51, with different HLA class 2 molecules for FAPA syndrome, and still different HLA molecules for recurrent oral ulcers. So HLA seems to have something to do with this as well. Another paper that we published has to do with the variants of the FMF gene that are associated with FMF, and they appear by population genetic studies to be highly selected, and actually these variants lead to increased responsiveness against bubonic plague. Finally, the last thing that we've worked on over the last year is an article that we published in the New Year's Eve edition of the New England Journal of Medicine, describing a new disease called VEXAS syndrome that is caused by somatic mutations in a gene called UBA1. Now, the interesting thing about this, and I don't have time to go in detail into how we did this, but basically we took a genotype first approach in which we had a list of genes that we hypothesized might have something to do with autoinflammation. We then used them to interrogate 2,500 exomes from undiagnosed patients. We found actually by using a number of criteria, 3 middle-aged men who were heterozygous for a particular variant of a particular residue of a gene called UBA1, which is an X-linked gene. And of course, this should immediately make you think, well, that's impossible because it's an X-linked gene, and I'm saying that these men are heterozygous for it. And in fact, what is the case is that they have somatic mutations in this gene that lead to the disease. Now that's, you can say, well, you know, that's interesting, middle-aged men, somatic mutations, it's similar to the idea of cancer. These middle-aged men also had on bone marrow vacuoles in their bone marrow. And from those vacuoles, we were able to identify other patients from our cohort and from a cohort of patients with relapsing polychondritis from NIAMS. And that led us actually to define this disease that's caused all of the patients by mutations in UBA1, and they are all of them men, middle-aged men. They all have somatic mutations in this gene. 40% of them have died, and they have a number of manifestations in terms such as sweet syndrome, pulmonary infiltrates, something that looks like relapsing polychondritis, venous thromboembolism, macrocytic anemia, myelodysplastic syndrome. And you can see that these patients actually were labeled with a number of different autoimmune diseases. But now that we know that they have mutations in UBA1, we classify them as having this new disease, VEXAS, which stands for vacuoles, E1 ligase, which is the type of enzyme that this is, X-linked autoinflammatory somatic syndrome. And I won't go into the molecular details on that, but just to sum up, the big picture of VEXAS is that using a genotype first strategy, one can define new illnesses based on genetic variants shared among patients carrying distinct clinical diagnoses. This may give rise to a new molecular taxonomy of rheumatic disease. Second, the somatic variants for genes encoded on the X chromosome are probably underrecognized. And thirdly, somatic mutation may account for a significant fraction of adult onset inflammatory disease. Well, with that, I'm a couple minutes over, so I've outstayed my welcome, but this is just a photograph of the clinical center of the NIH where a lot of this work was done and that made this all possible. Thanks a lot for your attention.
So Dan, let me first say that you have not outstayed your welcome, and we thank you very much for your presentation. Much appreciated.
Thank you.
If the committee members have clarifying questions, we do have a few moments, so please raise your hand feature. Use the raise the hands feature on the participants tab at the bottom of the screen, and I will call on you to speak if I recognize that from the screen.
We have a question from Michael and Emily as well. Both of them have their hands up.
Yeah, hi, Dan. That was great. One of the things the task of this committee is to talk about how NIH accounts for and does research in autoimmune disease. That's point number one. Number two is the genetic things that you talk about are in fact a small proportion of the totality of autoimmune diseases, though I suspect given another century, maybe all of the autoimmune diseases will fall into your category rather than that. How would you suggest we respond to the question of what is an autoimmune disease with your continuum from autoimmune into autoinflammatory?
Yeah, that's a great question, Mike. And you know, I think that at this point, we probably would want to include, you know, everything on the spectrum going from autoinflammatory to autoimmune, because I think that it's just that, you know, we want to think about disorders in which people have, you know, sort of unprovoked, or at least largely unprovoked. In a lot of these diseases, there can be some kind of environmental trigger, actually, but you know, disorders where you have some abnormality, genetic abnormality or acquired abnormality that someone may have that makes them more likely to have inflammation or frank adaptive immunity against their own tissue. So I would include all of those things, and I would just be ecumenical about the whole thing. In terms of your question of, well, how is it decided in the intramural program? At least I'll just speak to the intramural program of you know, who does what and whether people work on particular sets of diseases. At least in the Genome Institute, our policy is that really the decision point is at the stage of recruiting somebody, of hiring them as a tenure track investigator, because that's the point at which we're thinking about, well, do we want somebody that's going to be a bioinformatician, somebody that's developing new sequencing technologies, somebody that works in a particular human disease, et cetera. But once we have them there, then we feel that they should have the opportunity to follow their passion, whatever it may be. And unless they're not doing a good job, we're not going to tell them that they shouldn't work on X or Y or whatever it is. I mean, I suppose that if they started doing astronomy, we would have to step in. But otherwise, you know, we will just let someone follow their muse. And so it really is important then to decide who it is that you want, because you're making a commitment to that person potentially for, you know, several decades.
So I have Emily and Delisa on my list. I won't have time for anybody else. Two quick questions, one from Emily, one from Delisa, and we'll go on.
Hi, that was a fabulous talk. Thank you. One of the things you mentioned early on was about one of the features at the NIH would be long-term studies that require stable funding. I was wondering if you could provide a little bit more resolution on that. Would these include things like undifferentiated projects like cohorts, where you don't really know exactly what you're looking for at the outset? And I guess what I'm getting at is, what type of infrastructure do you see this driving that allows acceleration of hypothesis-driven research further on down the line?
Yeah, so you're right that the long-term kinds of studies can be following cohorts, and you know, sometimes it's a discovery-based kind of thing. I mean, I have a cohort of patients with autoinflammatory / autoimmune disease of like 3,000 patients, and I've been following some of them, or our team has been following them for, you know, up to 30 years in some cases. So, you know, you do get a real perspective in terms of the natural history and evolution of disease. In terms of then accelerating hypothesis-driven research, well, you know, there are different kinds of people, you know, some people that are more discovery based and then will collaborate on the mechanistic studies or the hypothesis driven studies, and then other people that are more, you know, mechanistic. And I think that, you know, one of the magical things about the intramural community is having that mixture of people, and having people, you know, that do well at what they can do to put it all together.
Delisa?
I think a lot of my questions were answered, but I guess one thought is with some of these investigators that really are looking at the genome using high throughput, where they have an opportunity to discover things related to autoimmune disease. Is there a mechanism for understanding what output might be relevant to different types of disease coming out from intramural funding?
Yeah, well, so that's a very good question. And, it depends on what the person publishes. Everyone is required to put out an annual report of what they find during the course of the year. So that's one way that the information does become public, even if it's not in a paper. But, I think that's a problem across the board in research, is getting all of these clues that, they're interesting clues, but you don't know quite what to do with them, and get them out there so that somebody else maybe can think about them. That's an unsolved problem, I think.
Thank you.
Well, thank you very much, Dr. Kastner. Extremely informative for the group. If anybody has any additional questions, put it in the chat and we'll try to get an answer from it, during the time of the meeting. Let me move on to Dr. Barron.
Good afternoon. I think you have control of my slides. There you go. So I'm representing the intramural program of the National Institute of Allergy and Infectious Diseases. I'm the Deputy Scientific Director, and if we can go to the next slide. So NIAID has a dual mandate to balance cutting-edge research in both the laboratory and the clinic, and has the capacity to respond quickly to emerging and re-emerging infectious challenges. Next slide. As Dan Kastner has mentioned, the intramural program has a relatively stable resources, and this allows the study of long-term and high-impact problems. And since our researchers don't have to continually apply for grants, and they are rated retrospectively, they can continue on their projects as long as the Board of Scientific Counselors approves of their or grades their programs as outstanding. One of the things of our program is that we were able to respond rapidly to public health emergencies, and I'll be talking about some of those. And again, our retrospective review by the BSc is unlike those in the extramural community. Next slide. We have our budget is about 7.5% of the NIAID budget. Currently, it's around $350 million. It changes, it seems, day by day by the additional COVID money that we have been given. We have 20 labs and branches. We have 133 principal investigators, maybe a total of 1,500 to 1,600 total staff members. And we have 44 research projects related to autoimmunity. And I'll be talking not about all of them, but some of them as I proceed. Next slide. We have laboratories in Bethesda and in Rockville. We also have a facility in Hamilton, Montana, where we have about 500 NIH contractors and employees. And this center has also houses a biosafety level 3 and 4 laboratory. We also have large programs across the globe. We have programs called ICERs, which are called International Centers for Excellence in Research. And this has an emphasis in Africa and in Asia. Next slide. So RDIR has been conducting research in immunity for over 60 years. Our longstanding commitment to the understanding of autoimmunity includes a number of very important advances. First of all, Tony Fauci of COVID fame described a revolutionary treatment for ANCA-associated vasculitis in 1973. Prior to this, about 90% of the patients had fatality within 2 years. In his study, 13 out of 15 had total remission. Bill Paul discovered interleukin-4 in 1982. And IL-4 is a master regulator of inflammation and underpins all current models of immunity. In 1995, Mike Leonardo identified autoimmune lymphoproliferative syndrome, or ALPS, as the first genetic disease of apoptosis. And I'll be mentioning this again in a few slides. And then Steve Holland in 2012 described the linkage of anti-interferon autoantibodies with adult onset immunodeficiency and opportunistic infections in Thailand. Next slide, please. As you know, there's an overlap in the spectrum of autoimmunity, immune deficiency, and autoinflammatory diseases. I threw up here some of the studies that are diseases that we study. I'd like to point out MISC, and I'll be talking about that in future slides. Next slide. While I can't detail achievement in all of our 44 programs related to autoimmunity, I'd like to highlight a few of our programs. Next slide. So primary immunodeficiency can present with a hyperactive immune disorder, such as autoimmunity, lymphoproliferation, and atopy. We have a primary immune deficiency clinic, which works to understand the immunologic mechanisms of the disorders. This clinic sees about 50 to 60 new patients a year, with another 200 to 300 in follow-up. We also recently formed the Blood and Immune Deficiency Cellular Therapy Program. It's kind of a mouthful, BID-CTP, which is an NIH-wide collaboration delivering state-of-the-art care for cellular and gene therapy. Listed below, or you see some highlights from some of the papers that have come out of this research. Next slide. Inflammatory bowel disease has several autoimmune and autoinflammatory features. Our investigators have a broad spectrum of basic and clinical studies related to the immune system governing the mucosal surface, with a few examples shown here and a few of the clinical trials. Next slide. ALPS, which I mentioned before, autoimmune lymphoproliferative syndrome. These patients have lymphoproliferation with large lymph nodes, liver, and spleen. They have a number of autoimmune sequelae, most commonly hemolytic anemia and thrombocytopenia. We have had a long follow-up of these patients, a 25-year-old follow-up, and are continuing to enroll patients in this natural history study. Next slide. APOSED, or autoimmune polyendocrinopathy, candidiasis, ectodermal dystrophy. Now, we may have to work with Dan to get better names for some of our diseases. Or APOSED is a monogenic disorder caused by AIR mutations, and these patients have chronic yeast infections and autoimmunity to a number of different organs, most commonly the endocrine system and the lungs. And we house the largest single center cohort of patients in the world with this disease. And listed again are two recent papers that have come out of this effort. Next slide. Dr. Golbach-Monsky leads our research in autoinflammatory diseases. She's been a world leader in identifying novel diseases and treatments. Next slide. Multiple sclerosis. So Dr. Bilyakova leads our research program in MS. A key point in her research is her data-driven machine learning approaches in the diagnosis and treatment of MS. One of her clinical trials will hopefully result in determination of biomarkers to predict which drugs a person with MS may respond. Okay, next slide. Lupus. So John Sang uses some of the newest advances to generate individualized predictions of disease risk and intervention efficacy. And Sylvia Boland has developed a mouse model of lupus. She's able to manipulate this model to understand cellular mechanisms that induce pathology. And she's also able to identify new genes as therapeutic targets. Next slide. We've also, over the years, developed a number of programs to support the research of our investigators. We have the Center for Human Immunology, Autoimmunity, and Inflammation. We also have a Center for Advanced Tissue Imaging and the NIH Microbiome Program, with examples of papers from each of these programs. Next slide. Now I'd like to take some time to discuss some hot off the press research on COVID-19. think everybody knows what that is, so I won't go into any further description of what COVID-19 is. Next slide. Next slide, please. Oh, there you go. So our scientists examined samples from about 1,000 patients with life-threatening COVID pneumonia and found that about 10% of the severe COVID patients have autoantibodies to cytokines. And these patients were predominantly men. And this is a paper showing that one of these antibodies was against interferon. Next slide. We've also initiated a trans NIH effort where we're studying MIS-C, where several autoimmunity features are highlighted, neutralizing antibodies, mechanisms of immune dysregulation, etc., as listed here. We're studying patients from Italy, Chile, Israel, and the US, and we're basically looking at the autoantibody presence and function, as well as a number of immune dysfunctions that we've been reporting. Next slide. As far as post-acute COVID syndrome, we have a few projects that are ongoing. One is a longitudinal study of COVID sequelae and immunity in adults at the clinical center. And that's been enrolling patients where a head-to-toe evaluation for long-term follow-up and involvement of different body systems is evaluated. We also have a longitudinal study of COVID sequelae and immunity in an Italian patient cohort. And we're soon to launch a longitudinal COVID-19 study in children, where we will be seeing about 1,000 children recovering from COVID, ranging from asymptomatic disease to MIS-C. Next slide. As far as existence of pre-existing autoimmune diseases, we have a project looking at a serosurvey of people living with rare diseases who are part of the rare disease clinical research. We also have a longitudinal study of COVID-19 sequelae and immunity in Italy that'll capture the changes in disease states in patients with ITP and other diseases. Next slide. So we've identified some gaps. One is the immediate need for COVID-19. We're looking at the vaccination efficacy in individuals with immune dysregulation. We're in the process of finalizing that protocol. And we're also finalizing a protocol of the study of allergic responses to vaccination. As far as the long-term needs, certainly recruitment and retention is a highlight of our gap. We'd like to augment preclinical research program and also have increased access to genetic computational biology and machine learning approaches. And with that, I'll be happy to entertain some questions.
Well, thank you very much. Certainly interesting and informative once again. I look forward to any questions, clarifying questions from the committee. Any hand raised, I will try to identify and answer. I don't see any. Awo, have I missed any?
No, I don't see any questions from the committee either.
Well, thank you very much, Dr. Barron. I look forward to further information specifically regarding the COVID studies, as we all do. And thanks again. Dr. Ryder.
Thank you. Okay, I'm Lisa Ryder. I'm acting head of the Environmental Autoimmunity Group in NIEHS, and I'm going to speak today about the NIEHS Intramural Program on Autoimmune Research. The mission of NIEHS is to discover how the environment affects people in order to promote healthier lives. And the vision of NIEHS is to provide global leadership for innovative research that improves public health by preventing disease and disability. Our main campus is located in Research Triangle Park, where most of our basic and epidemiologic research is conducted. We also have the clinical research unit in Research Triangle Park, where we see patients as part of our clinical studies, and the environmental autoimmunity group, which is part of the clinical research branch of the Institute. This is located in the NIH Clinical Center in Bethesda and is a unit focused specifically on systemic autoimmune diseases. Our interest in environmental factors really includes exposures early in life, pre-clinically prior to development of autoimmune disease, after signs and symptoms do develop, including ongoing disease, factors that may lead to disease persistence or even flare. We have studies related to basic epidemiologic, translational, and clinical research that include observational and mechanistic studies, and even clinical trials, such as patients we've studied with JAK inhibition that has improved their myositis disease activity. We study things through the lifespan, including in our unit, pediatric and adult disease. And we study both genetic and environmental risk factors for these diseases, as well as more broadly, disease activity and damage assessment, classification and disease improvement criteria, immune mechanisms of disease as well. Our intramural program research funding is structured very similarly to the other institutes you have heard from. Our NIEHS scientific director, who is currently Daryl Zeldin, determines the resources for principal investigators and bases this on comparisons with similar programs and productivities and the input of our Board of Scientific Counselor reviews. As you heard, these are retrospective reviews conducted every 4 years, looking at the research program's objectives, accomplishments, innovation, and relevance to the NIEHS mission, as well as their overall quality and impact. The laboratory and leadership is reviewed, and the performance of the research of the independent investigators. And over the past 2 decades, autoimmune disease research has been enhanced in the intramural program, with several branches now working on autoimmune disease. However, with the upcoming retirement of Fred Miller as the chief of the Environmental Autoimmunity Group, which I'm currently overseeing, the EAG resources will be decreased to align with similar programs in other institutes. To start with, we'll start with basic research conducted in our Immunity, Inflammation, and Disease Laboratory. In this laboratory, Michael Fessler, the chief, is leading research on immune-related GTPases, or IRGM, finding first that polymorphisms in this gene have been associated with autoimmune disease. He recently has completed work in mice null for the homologous gene LRGM1, which has developed a Sjogren's-like syndrome that it's driving excess interferon. There's infiltrates in the lung, salivary glands, and exocrine pancreas, which are blocked by knockout of interferon alpha receptor. And these mice also have high production of autoantibodies and cytokines. LRGM1 deficiency has been found to impair mitophagy, leading to mitochondrial DNA-dependent activation of cGAS and induction of interferon. So this suggests that defective autophagic clearance of mitochondrial DNA is an important cause of autoimmunity. The Environmental Polymorphisms Registry is a registry housed in the clinical research branch, and it has about 20,000 participants that have contributed biologic samples, as well as disease information and exposure data. And this repository is supporting a wide range of research on a number of different conditions. Michael Fessler's group has used this registry to look specifically at the IRGM polymorphisms, finding that there is an increased risk for Crohn's disease in one of these genes, and that reduced levels of IRGM and increased TNF is associated with the risk factor polymorphism. And he's further studying patients in the EPR registry to look at immunophenotyping as well as functional immune studies. He has future plans to do further knockout studies, as well as to look at immune pathways responsible for lymphoid aggregates in the lung, and to look at the profile activation status using CyTOF. from peripheral blood mononuclear cells. Another branch in our institute is the epidemiology branch, led by Dale Sandler. This branch is conducting a number of areas of investigation on autoimmune disease. First, the agricultural health study has about almost 90,000 licensed pesticide applicators who are mostly farmers and their spouses who have been followed over 25 years for cancer and chronic diseases. As part of this work, a subcohort have had samples and data to examine for autoimmune disease. Christine Parks, an epidemiologist working under Dale Sandler, has led studies showing that pesticides and other agricultural exposures have been associated with the risk of systemic autoimmune diseases, including rheumatoid arthritis, lupus, Sjogren's syndrome, and thyroid diseases. And specific pesticides have been linked to thyroid dysfunction and ANAs. There's ongoing work to look further in this cohort at some additional autoimmune disease outcomes. Another large study in the branch is the SISTER study, looking at more than 50,000 breast cancer-free women who have a sister who has had breast cancer. In these discordant sibling pairs, in addition to looking at breast cancer and other health outcomes, they have been studying autoimmune outcomes and found that early life risk factors for adult onset rheumatoid arthritis and lupus, including childhood residential pesticide use, and maternal and childhood farming and pesticide, have been risk factors for these diseases. And they have ongoing work to look at additional incident diseases, including thyroid disease, as well as studying the relationship of autoimmune disease to breast cancer. The Gulf study is another study of 32,000 participants looking at workers who were part of the 2010 Deepwater Horizon oil spill disaster, comparing them to workers who were not hired for the cleanup, but were trained. And this study is also looking at immune responses following exposure to oil spill chemicals, and it's including autoimmunity as an outcome. There are also studies in this branch looking at preclinical autoimmunity, including anti-nuclear antibodies in farmers in the agricultural health study. There's collaboration with the Institute on Aging, looking at the Baltimore longitudinal study on aging. Several studies have been completed. First, a study that has shown that leukocyte telomere length is shorter in patients with positive ANA, suggesting a defect in cellular aging associated with development of ANA or pre-autoimmune disease. And vitamin D deficiency has also been associated with positive ANA, and there's ongoing research looking at this. There has also been work in collaboration with our group looking at the National Health and Nutrition Examination Survey, or NHANES. Christine has led work showing that vitamin D deficiency has been associated with ANA in middle-aged and older adults, and is looking at factors associated with positive ANA in adolescents and young adults, a group of patients where there's increasing incidence of positive ANA over time, looking for factors that may be leading to this, including infections and asthma, phenol exposures, and parabens. Our own group, the Environmental Autoimmunity Group, has focused our work on autoimmune muscle disease, or myositis. We have developed largest databases and sample repositories in the world of these patients and led many national and international collaborations. Our work has focused on defining mutual, stable, and exclusive phenotypes based on the presence of certain myositis-specific autoantibodies, and these have allowed for the more precise characterization of risk factors, pathogenesis, and outcomes. We have done work to develop and validate myositis classification criteria and to develop and validate response criteria that are now accepted and endorsed by the American College of Rheumatology and the European League Against Rheumatism. Through these studies, we have been able to conduct multi-center studies of refractory patients looking at rituximab and also JAK inhibition as targeted new therapies. Fred Miller has developed the hypothesis that these diseases have different elemental disorders. That is that they have stable phenotypes that are recognized by cardinal signs and symptoms, pathologic changes, and molecular features, and that these come about by the interaction of multiple genes with environmental factors leading to disease, and the relative absence of protective factors that would lead to these diseases. And it's these interactions that are most important. So our group, Fred has led an international myositis genetics consortium, first performing studies of genome-wide association, finding that in more than 1,000 patients that the HLA 8.1 ancestral haplotype is the major risk factor for all forms of myositis, all of the major clinical groups and many of the major autoantibody groups. There are other genes that have been seen in association, including genes associated with other autoimmune disease, that is, genes that are involved in T cell signaling, general immune cell signaling, pyroptosis, and B cell signaling as well. And there are HLA risk factors for some myositis autoantibodies that are protective for others. Our group has also focused on environmental factors, and some of our key findings include that UV radiation is a risk factor for dermatomyositis and DM-associated autoantibodies, including MI2 and P155-140, also known as TIF1, both in the United States and globally. We've also studied smoking as a risk factor and found that this is a risk factor for polymyositis and also for Jo1 autoantibodies, including the anti-synthetase syndrome. HLA-DR beta 103, the primary risk factor allele in myositis, further increases the risk of smoking for these subgroups. We also have found factors associated with disease flare, including some of the same factors we see at disease onset: sunburns, infections, and certain medications to be associated with disease flares. Our ongoing work is continuing to look at novel genetic and environmental factors and the interactions of these. We have studies looking at twins and siblings discordant for systemic autoimmune disease. We have a study in early onset anti-synthetase patients and a study of military patients who have developed myositis while under active duty military service who have had some unique exposures. We've continued our work on detailed assessments and phenotyping and trying to work on novel therapies for calcinosis and soon for dermatomyositis. We also have established a national myositis patient registry with the Myositis Association. This study has enrolled 1,800 patients with different major clinical groups of myositis. We have found quality of life significantly impaired compared to a normative population and even to rheumatoid arthritis patients. Christine Parks led work looking at UV radiation exposure and found that job exposure in men and hobby exposure in women, these personal exposures, are risks for dermatomyositis. And we're continuing to work on this study looking at occupational and hobby exposures, infections, and medications, looking at different risks for different phenotypes of disease. The Environmental Polymorphisms Registry, which is housed in the clinical research branch, has continued working also in the area of immune-mediated diseases. They have combined a number of immune-mediated diseases, including autoimmune, allergic, and asthmatic conditions, and have found through detailed analysis that the PTP-N22 SNP, which is a risk factor for a number of autoimmune diseases, is a risk factor for these combined diseases, and that there are SNPs in the aryl hydrocarbon receptor as well that are risk factors for autoimmune disease. And they're continuing to work on additional genetic and environmental risks and their interactions. Fred Miller has led a large population-based study from NIHs looking at the prevalence of positive anti-nuclear antibody in the general population, showing that there's an increasing trend with age and highest risk in older individuals and in females. And there are also more disease-specific autoantibodies, including rheumatoid factor, antithyroglobulin or thyroid peroxidase, and tissue transglutaminase that are specifically present in these older individuals and in females. He also has shown that the ANA prevalence has increased over the decades from 11 to now 16% overall. And these risks are particularly high in men, in adolescents, and in white individuals and older individuals. And this is continuing to be studied to look at additional risk factors. In terms of our future wish list for other autoimmune disease studies, we would feel that looking at multidisciplinary consortium among basic and clinical research to optimally utilize databases, registries, repositories, and even animal studies would be helpful. To integrate autoimmunity as an outcome in many of the NIH cohort studies and registry studies, and even to create a national autoimmune disease registry would be of great interest for further work in the environment and in genetics. We think that detailed clinical, environmental, and molecular assessments of high-risk populations would be productive, including patients who have been in high exposure settings or patients with multi-case families, and that studying things over the lifespan and even pre-disease to disease progression would be quite helpful. We also think that detailed phenotyping and environmental exposure studies coupled with detailed immunologic and molecular phenotyping and genetics would be very helpful. And then we think that there should be increased attention to exposure assessments and biomarkers, environmental sensors placed throughout the US in targeted areas, enhanced geographic information systems to look at modeling and mapping of exposures, increased attention to wearable real-time personal sensors, and a new approach called the exposome to look at a number of lifetime exposures from conception onwards and to look at a number of these, looking at wearable sensors like silicone wristbands, estrogenic activity, stress, immune modulators, looking at measuring hormones, antibodies to pathogens, xenobiotics, and proteins, and looking at gene expression and epigenetics. This type of study has been tried. An EWAS study, environment-wide association study, tried in Niehs cohort with type 2 diabetes, looking at environmental risk factors. EWAS, I would say, is the GWAS of the environment. And this study in particular looked at a number of different exposures, including allergens, microbes, smoking, hydrocarbons, heavy metals, pesticides, plasticizers, and others, and found certain risk factors looking at all of these simultaneously, including certain nutrients that were protective and some that were risk factors, one pesticide that was a particularly strong risk factor, and PCB as well. So this type of approach could be very powerful to identifying additional risk factors. So we think that enhanced studies of autoimmune disease, that new approaches to studying and new technologies to carefully define these diseases and their phenotypes, to identify environmental and genetic risk and protective factors would help lead to the development of new disease pathways and better treatments, and ultimately help us to prevent these diseases by our better understanding of these factors. I left you with the resources from our institute, including our website, strategic plan, and the laboratories and branches that are currently conducting research in autoimmune disease. Thank you for your attention.
Thank you so much, Dr. Ryder, for the presentation. Extremely, once again, helpful to our committee. I do believe that Dr. Fred Miller is also online. So as we go through the brief clarifying questions from the committee, I'm sure Dr. Miller will be happy to answer any that are particularly directed to him. So any questions from the committee? I do see that Emily has her hand raised, and so does Glinda. So let me go to Emily first and then Glinda.
Hi, thank you for that wonderful talk. And your group has been doing such interesting work. One of the things I was wondering about, you mentioned as a wish list item, including autoimmunity as an outcome on large cohort and registry studies. Your group has successfully leveraged Niehs, for instance, in order to look at ANAs. But as I'm sure you know, most of the autoimmune outcomes are lacking in these sorts of databases. So what, in your opinion, would it take to increase data related to autoimmunity or autoimmune diseases in some of these national or NIH-driven cohorts and registries? Are there specific ones that are in existence that you think that data collection should be augmented, or would you see these as more de novo cohorts that would need to be started?
I think going forward, as new cohorts are developed, it's good to lay out that autoimmunity should be one of the outcomes captured prospectively, so we don't have to go back and guess about these things. But I think even retrospectively, one could look at ANAs, as was done in the NHANES cohort, as a starting place. And then if there were a possibility to go back and get follow-up on patients, then you would be able to probe more deeply. As is being done in the agricultural health study, they have gone back to ask additional questions about autoimmune disease. So I don't have a specific cohort in mind, but I think we need to put on the table that that should be a key outcome in future disease cohorts, especially large ones.
And oh, I'm sorry, can I just ask one further clarifying aspect to that? Would you envision these being cohorts run it from your group or in other areas of NIH that would require some sort of collaboration to make sure this gets on the radar?
I think that these are mainly large cohorts from other groups. For example, several years back, there was a desire to have a national children's health study, and I was in a committee there and spoke out for the need to have autoimmune outcomes. You know, we're not always at the table, so something from your committee directing this to other people might be helpful. But I think we also are continuing to have large cohorts of myositis patients. We're also happy to collaborate with this work when these questions come up.
Linda?
Hi, Lisa. Thanks so much. That was a great talk. And it's so nice to see all the work you're doing. Hi, Fred, wherever you are. I just have a couple follow-ups. And you're in such a unique institute, because you do have this environmental focus. And so you mentioned the environmental polymorphism registry and that it includes some kinds of environment measures, but the work that you talked about, specifically the examples, it did seem it was genetic. It was not yet incorporating environmental interactions. So I wanted to know, what was being measured, and if you're, even within your institute, if there's barriers to getting the crosstalk between the environmental exposure people and the people coming at it from a different direction.
Fred could also answer. I think he's been more involved. He's been on the steering committee of that group, so perhaps he should take that.
Hello, everyone. I'm Fred Miller, currently Deputy Chief of the Clinical Research Branch in NIEHS and about to retire and become emeritus, actually. I do think that there are limitations in communication within groups, within institutes, within the NIH, and across the country and the world. And that's one of our big challenges right now, is just having people talk with each other to know what's going on, know what their plans are, to know what the possibilities are, and to optimally utilize all of the resources that really are out there right now, which many of us don't know about. And or creating some sort of central catalog, perhaps, or even international registry of various resources like this, so that when people do develop a question, they could perhaps go to this registry and then do a search for what they want and find that, someone's already collected 1,000 patients with this condition. This is a challenge right now, I think, for all of us right now. And to answer specifically your question, trying to link the environment and the genetic aspects, you're right. There is no Institute of Gene Environment Interaction. There is an Institute of Genetics, there's an Institute on the Environment, and there's institutes focusing on specific diseases. But seldom do they communicate or optimally interact. That's one of our other big challenges right now, both because of the mandates from Congress as well as from our decisions to divide off certain information and certain proposals into certain institutes. So I think cross-institute RFAs, or requests for applications relating to these specific questions, when they are already well-established cohorts perhaps that have already established genetic and or environmental data that could be amplified that way would be a very cost-effective way to begin to answer some of these questions. So I think you're right, Linda, there are challenges here but there are ways to address them.
Another, I think what really came out of your presentation is that collaboration that you have with the Intramural Epidemiology program. And it's most of the people I think are doing population-based epidemiology as opposed to clinical epidemiology, which I think many of the other institutes may, maybe that's where their emphasis is more than the population-based epidemiology. So I do think that's a really big strength too. And well, I'm not going to put words in your mouth, but I assume that that's been a productive collaboration. There is, I know I had a question, and I think other people have brought up, wondering about the relationship between intramural and extramural, and does your work, like how is it shaping maybe RFAs or the communication between the two different branches in terms of thinking about the directions for future research at the Institute? I don't know if you want to talk about that, or maybe we could leave that for the panel discussion at the end.
Yeah, Mike Humble is going to address some of these issues where we work closely with him and some extramural collaborators, and he'll present some of that work, and that's probably a discussion better left for later on.
I might just specifically say for NIEHS that Fred worked very closely with Mike on several workshops that brought in experts and reviewed a current state and directions for future research. So there was a series of papers, and that certainly shaped a lot of focus for extramural program.
Well, thank you very much. There was one question that came in earlier from the audience, not from the committee, related to research related to mind-body connection and inflammation. I thought that we had a few moments, perhaps somebody on the panel would be happy to address that issue.
Well, I'd be happy to take that question. We are currently planning a study, and it's a little early to speak about, but it will be a clinical trial in patients with dermatomyositis, and it will be looking at taking in information about their various lifestyle factors through a phone app, and also doing some monitoring, and then trying to alter those through behavioral health coaching approach. So, and then looking at the effects on various immune pathways and so forth. So I think it'll be a very interesting study. We're in the planning phases.
Thank you. Well, seeing no more hands raised, thank you both very, very much for the presentation and for the questions thereafter. Appreciated by the committee, myself, and I'm sure the audience. Dr. O'Shea.
Hi, I'm just turning on my camera and okay, can you see me and hear me?
Yes.
Good. And it looks like you're going to run my slides for me. That's your preference? Yeah. Looks like they're up.
Alex will do that for you.
Okay, sounds good. So first, thanks very much for inviting me to participate. This is really terrific, and I very much appreciate being invited. One thing, a small correction. Thank you for the PhD. My wife will be very impressed. I think she'll think I'm smarter, but I'm an MD. If you could just note that, and it'll become clear as we go through my clinical interests. The other thing I was going to say is I looked through the slides from some of the earlier presentations, and I think you're going to hear a number of themes that are pretty similar that I'm going to try to articulate that you've already heard before. So hopefully I'll say a few things that haven't been touched on, but just giving you the big picture, focusing on NIAMS. So NIAMS, as I think you may know, is about 1.4% of the total NIH budget. And the intramural program of NIAMS is 11% of that 1.4%. But that means that it's about 1.4% of the total NIH IRP budget. And so on one level, you could say we're pretty small, but I think, as you'll see, we consistently punch way above our weight class and have an impact on the field. But in essence, that 1.4% basically means we have about 20 principal investigators, and that encompasses 11 tenured senior investigators, 4 tenure track investigators, 4 assistant clinical investigators, and 1 contract orthopedic surgeon. So NIAMS IRP covers a fairly broad swath of medicine, from rheumatologists, dermatologists, to neurologists, and we're focusing on diseases related to muscle, bone, skin, immunology, and then basic considerations: structural biology, chromatin regulation, and gene transcription and RNA biology. And this has probably been touched on by others before, but the way it works in the intramural program is that these investigators listed get reviewed every 4 years. And particularly with more senior investigators, more established investigators, the review is largely retrospective. So in that sense, it's rather different than prospective grants, where people are being judged by what they're going to do and how they're going to do it, and this sort of thing. So it's more about what you've done. And so as a net result, we basically like other mechanisms at the NIH, invest in people who are doing the work. But for us, it's very much, the emphasis really should be on long term. So we invest for people in many cases for decades, or for their entire career at the NIH. So a question came up was, well, about your strategic plan. How do you think about leading the institute? And in many respects, that the way you lead the institute is to think hard about who you're going to hire. Next slide. And so a little bit of history. So the National Institute of Arthritis, in a way, was started in 1950 by Harry Truman. And one of the people who was hired at the time was Joseph Bunham, who was the clinical director at that time. And he made rather important inroads into understanding arthritis. And one was a clinical test for rheumatoid arthritis, shown in the above panel. But equally, if not more important, he was really one of the first people to use what's now the gold standard drug for treating rheumatoid arthritis, methotrexate, way back in 1964. And this was his sort of landmark paper at that time. Next slide. In 1985, I think it was Barry Goldwater who pushed to have an institute, an institute of itself with arthritis, but also in the end, when it worked its way, I guess, through Congress, there's an Institute of Arthritis, Musculoskeletal, and Skin Diseases, and that's the present institute in 1985. But NIAMS at that time picked up, in a way, what Joseph Bunham had done with cancer drugs, and thinking about how to use those drugs to treat autoimmunity. The NIH really became, really led the field in generating new treatments for lupus nephritis. And that was the use of drugs like cyclophosphamide with or without steroids, and I'll come back to that in a bit, in various combinations over the years, starting in 1986, and then a number of follow-up studies. Next slide. The scientific director, the first scientific director of NIAMS, was this guy, Henry Metzger, who really was so much the scientist. And at that time, he was very interested in cell signaling. And I have this sort of cartoon on the right, which depicts that he was interested in FC receptors, which are, of course, very pertinent. And he was mostly interested in allergies. But as I think many of you know, FC receptor is very important for other immune-mediated diseases, including lupus. And he was one of the people who cloned and really discovered and described the structure of FC receptors. And they very much became a paradigm for this whole class of multi-chain immune recognition receptors, which includes things like the T cell receptor, B cell receptor, on and on and on. And so really was a giant really in his field scientifically. And as we'll get to in a moment, his obviously insights were sort of led me to indirectly, you'll see, to do what I did. But equally important, Henry had the insight to hire two people I think have made an impact in NIAMS, me being one of them. And the other person, next slide, was Dan Kastner. And I won't reiterate all of the things Dan did, but both of us were hired to work for Henry. And then Steve Katz appointed Dan and I as Dan as clinical director and me as scientific director in NIAMS. So before Dan went on and went to the Genome Institute, he helped direct what we've been trying to do over the last 15 years or so in the Arthritis Institute. Next slide. So what I was thinking about when Henry hired me was we're in this midst of a revolution in a way of the sort of the molecular biology revolution for immunology, where pretty much every week it seemed like you were discovering a new cytokine, cloning in another cytokine and a cytokine receptor. Next slide. And that was, people capitalized upon that very quickly to make all of these new drugs that target different cytokines and different cytokine receptors. And for a person like myself who sort of witnessed the early days of treatment of a variety of diseases from lupus to rheumatoid arthritis and my own area of interest at the time when I first came to the NIH and worked with Tony Fauci, the drugs for the treatment of lupus with cyclophosphamide, the drugs were efficacious, but they had lots of toxicities. For rheumatoid arthritis, a very difficult disease to treat at the time, but then it was almost miraculous seeing how biologics had an impact on these diseases. And again, that's the other part that I wanted to emphasize as we go along, is the importance of the basic science that as we discover all of these different molecules, it's just astonishing to me how quickly and efficiently you can generate new treatments once you figure out who the players are and how they relate to disease. But nonetheless, with this class of drugs, even though they're really quite extraordinary, there are diseases that these drugs don't work with. And even in diseases like rheumatoid arthritis where they do work, a substantial number of patients don't have complete responses. Next slide. Next. Sorry, I have some animations here. That's great. So despite the so despite the success of biologics, as I was saying, not all patients respond. They don't respond completely. And so at that time, we were thinking, well, what other options might we have? And we were thinking, well, what if we learn more about how cytokine signaling, following on Henry's insights, and can you use that knowledge to target molecules therapeutically? And Lisa already referred to this, that this led to the discovery of JAK inhibitors, which I'll tell you about the discovery of that in the next slide. And so we and others were trying to understand how these cytokines worked, especially the class 1, class 2 cytokines. And what we and others did was knowing, again, based on what people like Henry Metzger had discovered, that cytokines were an early step in cell signaling. And we and others found the small class of kinases called the Janus family kinases. And as I'll show you, a number of groups figured out which JAKs work with which cytokines, et cetera. And another former NIHer, Jim Darnell, who was at the Rockefeller at the time, took a different approach where he was thinking about interferon signaling, and instead of sort of blindly trying to figure out who were the partners that were activated with these different cytokine receptors, he looked at the DNA and then used that as a bait to find this new class of transcription factors called the STATs. So it became clear pretty quickly that there's about 57 or cytokines or so that use this pathway, the JAK-STAT pathway, for signaling. But at the time, we didn't know really. We know a lot about what was happening in cell lines, et cetera. But really, what was the in vivo criticality of different of JAKs? And next slide. The discovery, in a way, you might imagine that the way people would do these sort of experiments would be make knockout mice, et cetera, et cetera. But we came to the discovery of what they actually do a little more quickly, and that was sort of the result of work from a number of people who are shown here on the right-hand side of the slide. And basically, the way the story went was that when we cloned JAK3, we cloned it from lymphoid cells, and we were suspicious then that this kinase was important for signaling via cytokines that are important for lymphocytes. And I had been a postdoc with Warren Leonard up at the top here, and Warren had just around that time had discovered that mutations of the common gamma chain shown here underlies X-linked severe combined immunodeficiency, basically bubble boy syndrome. And because of the expression of the common gamma chain, we hypothesized that this new Janus family kinase, JAK3, is associated with the common gamma chain. And we showed that with Warren. And in that paper, we hypothesized that maybe there would be children who had mutations of JAK3, but had a phenotype quite similar to X-linked severe combined immunodeficiency. And so maybe, girls who looked like they had the disease that boys had might have mutations of JAK3. And so another NIH alum, Gigi Notarangelo, in Brescia, Italy, Gigi has returned now to the NIH and is in NIAID, with Fabio Condati, who actually had then moved to my lab. Gigi looked in his cohort of patients, and as did Rebecca Buckley at Duke, and found, in fact, that our hypothesis was correct, that there were children who had a phenotype similar to X-linked severe combined immunodeficiency and who had mutations of JAK3 instead. And so instead of making a knockout mouse, we benefited from studying human immunology and identified the first pathway in the JAK-STAT pathway. And so this is another example that I wanted to use to illustrate the point that what we often do at the NIH is to study rare diseases, but they have broad impacts for more common diseases. And the way that this has an impact was that we speculated at the time that if you purposely made a JAK inhibitor, that would be a new class of immunosuppressive molecules. And so, in fact, we did that. I had actually, when I first cloned JAK3, I had met at a meeting a Pfizer scientist, and we started a collaboration actually 2 years before this in 1993. And we did this through a mechanism called a CRADA, so that we've basically had a collaboration with Pfizer since 1995. Next slide. And now we have 9 JAK inhibitors. I'm not going to go through all of the details for you, but simply point out that there are JAK inhibitors approved for hematological indications, including acute graft versus host disease. That's ruxolitinib, a JAK1, JAK2 inhibitor. Tofacitinib is the Pfizer compound, and it's approved for rheumatoid arthritis, psoriatic arthritis, juvenile arthritis, and also ulcerative colitis. Baricitinib is the is the Lilly compound. It's approved for rheumatoid arthritis in the US and atopic dermatitis and rheumatoid arthritis in Europe. Pefficitinib is a drug that's approved in Japan. Fedratinib, another JAK2 inhibitor. Upadacitinib is one of the first sort of second generation JAK inhibitors that has a bit more selectivity for JAK1 over the other JAKs, approved for rheumatoid arthritis, also approved for psoriatic arthritis and ankylosing spondylitis in Europe. Filgotinib is a selective JAK1 inhibitor approved in Europe. Delgocitinib is a JAK inhibitor, topical JAK inhibitor approved in Japan. And believe it or not, there's actually a dog JAK inhibitor as well, that's oclacitinib. Next slide. And again, this just illustrates the number of ongoing trials in a wide variety of diseases with JAK inhibitors, ranging from classical autoimmune diseases shown here, different arthritides, and also dermatomyositis, but also for inherited interferonopathies, which Dan, I'm sure, talked about, and other autoinflammatory diseases. And it's now recognized that Down syndrome, for example, that with trisomy 21, some of the genes that are amplified are interferon receptors. And so Down syndrome is considered an interferonopathy, and that's being studied for JAK inhibitors being studied in this disorder. Many dermatologic diseases as well that are listed here. I'm not going to go through the details. I mentioned inflammatory bowel diseases, but also in the setting of oncologic indications. And one thing I neglected to say on the last slide, but don't go back, is that the Lilly drug, baricitinib, is also has emergency authorization for COVID in COVID cytokine storm. Next slide. So just to review where we are at the present time in NIHS, I'm going to quickly go through some of our areas of current investigation. As I alluded to, there are labs that mostly focus on basic problems from epigenomics to chromatin biology and RNA biology, gene expression, cell biology and development of, in this case, of skin cells, structural biology, and a lot of immunology. We're still focused on lupus many years after the work that was done when NIHS was first founded, with hiring Mariana Kaplan. And Mariana's interest in lupus largely relate to identifying the role of neutrophils, but also we're interested in genetic aspects of lupus. Mariana is interested in how lupus and cardiovascular disease relate, and I'll try to come back to that in a bit. We've recently hired another investigator who's interested in the action of steroids. And again, maybe hard to imagine, but there's still a remarkable amount to be learned about how steroids work, even though they were first discovered and used for therapy in the 1940s. So nearly, 80 years later, it's really quite shocking to realize what we don't know about steroids. We're using JAK inhibitors, as you might imagine, in a number of disorders. Mariana is also very interested in metabolic regulation and is interested in drugs like PPAR gamma and the treatment of lupus. But we're also involved in collaborations looking to see how diet affects lupus and how diet and microbiome interact. Interested in Sjogren's syndrome, including the utility of JAK inhibitors. Big interest in vasculitis, Peter Grayson's work. You heard about Dan's and Peter Grayson's collaboration, the discovery of the disease Vexus, somatic mutations, spondyloarthritis, and particularly juvenile spondyloarthritis. Our investigators, Andy Mammon, is interested in understanding myositis, the mechanisms, but also therapeutic interventions. We're interested in juvenile arthritis, and again, particularly interested in the genetics of juvenile arthritis, similarly scleroderma, genetic and epigenetic impact on scleroderma. Next slide. As I mentioned, part of NIAMS is dermatology, and won't spend a lot of time going through this, but we're also interested in orthopedics. That's part of what we do. We have a small sort of research initiative here with our contract orthopedic surgeon. For a long time, we've been interested in viral structural biology. And also we have a dentist who works on our institute and is interested in regulation of bone and a dental oral craniofacial development. Next slide. Okay, so just to start wrapping things up, I thought I'd touch on a couple of points. So I was thinking about, thinking about autoimmunity broadly and thinking, is this word still useful? And obviously on one level it is. That's on one level a foolish question, but at the same time, the reason I wanted to pose that question is Dan Kastner's talk before is that when Joe Bunham and Henry Metzger were doing their work, we didn't even use the word innate immunity. That term hadn't been coined. We obviously knew about things like neutrophils, but if you read books from that era, they're fairly dismissive and say, neutrophils are, non-specific immunity. They didn't really think hard about how innate factors and adaptive factors interact. And I just give you an example here is that, we know that lupus patients have fever, but not a lot of focus on mechanisms to this back in the early days. People obviously were interested in mechanisms of fever, but putting it together of innate and adaptive mechanisms was not something people thought a lot of into the 1970s, 1980s. And Mariana Kaplan is another good example that she focuses on neutrophils in lupus, and it's not something we spent a lot of time thinking about in the 1980s. And what's particularly interesting to me is that Mariana, as you know, is that one of the big morbidities associated with lupus is cardiovascular disease. And so Mariana puts it all together and sort of thinking about how adaptive mechanisms, innate immune mechanisms affect things more sort of common problems like coronary artery disease. And so I'd sort of emphasize that now as immunologists, we really sort of think of all cells are immune cells. There's no cells that are not immune cells. All cells have host responses to infections. And so in thinking about these diseases, we have to be much more holistic. And the converse of that is that a common disease, coronary artery disease, we know that an inflammation is a big part of that. So that elevated CRP, of course, is an independent risk factor for coronary artery disease, independent of other classical risk factors like lipids and this sort of thing. And of course, then conversely, drugs like colchicine, which we use to treat inflammatory diseases, can be used to prevent heart attacks, et cetera. Equally, there's more recognition that metabolism and inflammation are intertwined, that the ways that immune cells get activated and the metabolic changes are not trivial. They're unique to different cells, et cetera, et cetera. And one of the things we'd like to do, as I mentioned, is to have a protocol with dietary restriction in patients with lupus. And this is something we're planning with NIAID scientists, including Yasmin Belkane. Next slide. The other thing that I was thinking about, and I'll give you an example from my work, but this is really that a lot has been done by AMP, the Accelerated Medicines Partnership. And again, it gets to this issue of silos that immunologists work on immunology and endocrinologists work on endocrinology, et cetera, but really all of those specialties in a way are misleading in the sense that if you're an immunologist and you discovered a factor, you called it a cytokine. If you're an oncologist, you discovered something, you called it a growth factor. But in fact, all of these factors are all about cell-cell communication. And I'll just give you an example. We did an experiment that's sort of illustrated on the right. I'm not going to go through all the details, but we challenged mice with a parasite, and then we looked to see what happens to the immune cells in the lungs of these mice. And suffice it to say, we were shocked to find that a class of innate lymphocytes express this gene, calc-A, which is actually represents a transcript that encodes not calcitonin itself, but a related product, which is CGRP, which is the thing that drives migraine. So in essence, what we didn't know is that when mice are challenged with this parasite, they basically get migraines in their lungs, and the cell that's making this product is an immune cell, not a neuron. And not only that, that actually these innate cells and adaptive cells both respond to this neuropeptide, and they respond to other neuropeptides as well, including neuromedin. So the long version of this is that the nervous system, the immune system talk to each other. And again, some of these words are misleading, that I guess they're neuropeptides, but if we had discovered CGRP a long time ago, we would have called that a cytokine. And the other thing, there's people like Brian Kim from WashU who have very nice work showing that neurons respond to cytokines. Neurons have cytokine receptors. So when you're thinking about problems like pain, itch, fatigue, you really should be thinking as well about this more holistically, that the cells are all talking to each other. And again, discussion of the Accelerated Medicines Partnership, that's a separate discussion, but again, using single cell technologies is really important in moving beyond sort of simplistic views of immunity. And again, the message for me is that single cell RNA-seq really is something that breaks down silos. And another example of this is how we now know that rheumatoid arthritis really starts in the lung. So and smoking obviously is a big risk factor for rheumatoid arthritis. So rather than just focusing on the joints, we have to, again, think more broadly. Next slide. And again, I realize I'm saying things that I think you heard from Jane Buckner and others earlier in this meeting. I did want to go on, I'm sure Dan went in great detail about this, but just again, thinking about genetics and thinking about genetics more broadly, we obviously know about genetic risks of a variety of immune and inflammatory diseases, ranging from single gene diseases, and then Dan, no doubt, talked about somatic mutations. Lisa was talking about thinking about genes and environment, the importance of that. Obviously, when you're thinking about genetics, you can't just think about our genetics, but also the genetics of the organisms that populate us. And then in sort of thinking about genetic risks, it's not just thinking about genes. One needs to think beyond conventional genes, and again, thinking more broadly what Waddington was thinking about. And again, let me just give you a short example. Next slide. When we were first doing these experiments on the JAK-STAT pathway, I illustrated something that's very much a 20th century view of the genome, which really focused on genes, classical genes, and promoters. But what we know, thanks to our boss, Francis Collins, is that only 2% of the genome is genes and 98% of the genome is not, but it's not junk. It turns out that much of the genome is transcribed. And so transcription is way more than just, as illustrated to the left, with transcription factors binding to promoters, that basically when you activate cells, you're influencing the entire genome. And so much of the genome is activated in cell-specific and activation-dependent manners. And we have thousands of cell-specific enhancers. And of particular interest are regions known as super enhancers. We have high density of active enhancers. And often in these areas where you have active enhancers, density of active enhancers, you have things called long non-coding RNAs. And in fact, the number of long non-coding RNAs probably exceed the number of conventional genes. And so in thinking about the genetic links to human disease, you have to think about way more than the 20,000 or so conventional genes we have. You have to think about the regulome and also non-coding RNA. And I'll give you one example of that from, again, from my own work. Next slide. And I'll just show you a really quite heroic experiment that we did in collaboration with Rafi Ahmed. So Rafi took individuals who were vaccinated with yellow fever vaccine and remarkably followed them for 10 to 14 years. And he looked at primary responses, but then looked at responses 10 to 14 years later. And suffice it to say, what this experiment shows is looking at a non-conventional gene, the interferon gamma antisense transcript. So it's a transcript that is encoded within the broader interferon gamma locus. So interferon gamma is a cytokine, but it's transcribed backwards in a way. And when Rafi looked at his antigen-specific cells 10 to 14 years later after the vaccination, what he found is that this antisense gene, this non-canonical gene, long non-coding RNA, is expressed at high levels in these antigen-specific cells, even though the product, the nearby product, the interferon gamma locus, is not expressed. So next slide. So in thinking about this, obviously we have a long way to go, I think, in how we benefit from the therapies that we're using. So no question that targeted therapies are amazing advances, but I think we will continue to have the need for combination therapies. Even now, even with drugs like cyclophosphamide, they're used with steroids. But we want to use those in the smartest way we can. And so I've given you an example here on the right that biologic DMARDs are a bit like a light switch, that we have tendency to turn things on, turn them off. Targeted synthetic DMARDs like JAK inhibitors, we can titrate the doses better, but mostly we don't really do that. We identify a drug that's safe and effective, a dose that's safe and effective, but we don't really modulate the doses, and we don't use them modulate the doses with biological DMARDs. So I think we have a long way to go to really understand the pathways by understanding the genetics better, by measuring things better, and then to appreciate individual variation, heterogeneity disease, and the different phases of diseases, et cetera, et cetera. So we have a long way to go with that. And so I think I'll stop with that. And I apologize, I ran over a few minutes.
Well, thank you very much. Thank you so much for your presentation. Very helpful to us, as we've had a lot of issues related to your comments. I'm going to hold questions at this moment for Dr. O'Shea until the end of the presentation, when we have a larger Q&A, since we're scheduled for a break just about this time. So Dr. O'Shea, thank you once again. I hope you'll be here at the time of our Q&A so that those questions that were perhaps raised now can be answered later. We will take a 15-minute break at the moment and come back at 3:45 before proceeding to the next part of our meeting. Once again, thank all the speakers and all participants, and we'll be back in 15 minutes.
Hi, it's Lisa. Are we ready to go? I'm just wondering.
I'm just waiting for, yep, he's just opening up now.
Okay, great.
Welcome back, everybody. I think it's been an extremely stimulating first half of the day, and I'm looking forward to the next. For the second half of today's panel, we'll hear from representatives from the 3 institutes at the NIH that fund extramural autoimmune disease research as opposed to the intramural of this morning. The National Institute of Diabetes and Digestive and Kidney Disease, the National Institute of Environmental Health Sciences, and the National Institute of Minority Health and Health Disparities. Once again, each panelist will offer 20-minute remarks on their IC's focus on autoimmune diseases and any changes or any evolution in funding priorities or topics for research, significant achievements or progress from the funded work, and existing research gaps, all of which are very interesting and important for our committee work, and some insight into the strategic planning for identifying areas of focus for solicited research funding. The format will remain the same, and however, after all of the remarks, we will have approximately 30 minutes or so for discussion with all the panelists, and that will be helpful again in clarifying and informing us further. Let me introduce all the speakers now. Lisa, and by the way, the bios are available. Lisa Spain is the program director of the Immunobiology Type 1 Diabetes and Autoimmune Endocrine Diseases program in the Division of Diabetes, Endocrinology, and Metabolic Diseases at the National Institutes of Diabetes and Digestive and Kidney Diseases. She's also the project scientist for Type 1 Diabetes TrialNet, an international consortium that conducts clinical trials for the prevention and reversal of Type 1 diabetes. Her special interest is facilitating biomarker discovery. Michael Humble is a toxicologist, and he is a program administrator in the Genes, Environment, and Health branch of the Division of Extramural Research and Training at the National Institutes of Environmental Health Sciences. In addition to overseeing fellowship and summer research programs and an undergraduate research education program to enhance diversity in the environmental health sciences, he oversees the research portfolios in autoimmune diseases, immunotoxicology, and skin disease. Dr. Nishadi Rajapaksha is a program director in the Division of Scientific Programs at the National Institutes on Minority Health and Health Disparities. She directs the Transdisciplinary Collaborative Centers for Health Disparities Research focused on the precision medicine ideas and initiative. An initiative to better understand the complex interplay of biological, behavioral, social, environmental factors in developing new diagnostic or therapeutic approaches. She also leads the Centers of Excellence on the Environmental Health Disparities Research Program, which is a joint undertaking with NIEHS and EPA to investigate environmentally driven health disparities and also to improve access to healthy environments. She is also a program official for the R01 program and the research centers in minority institutions. So again, we'll have a few moments, I hope, for clarifying questions after each speaker. And let me start with Dr. Spain.
Great. All right, thank you so much. Again, my name is Lisa Spain, a program director for autoimmune endocrine diseases and project scientist for Type 1 Diabetes TrialNet. Next slide. The National Institute of Diabetes, Digestive and Kidney Diseases mission is to improve health and quality of life for people with diseases related to these organ systems. Autoimmune disease research is funded by all 3 divisions, AUH, Kidney, Urologic, and Hematologic, DDN, Digestive Diseases and Nutrition, and my own division, DEM, Diabetes, Endocrinology, and Metabolic Diseases. My training is in Immunology. Next slide. This slide shows an overview of current active awards as identified by the publicly available NIH reporter system. This wheel that you see here is similar to a word cloud. It's not really quantitative. The same grant can appear in more than one of the quadrants, but it illustrates the broad categories of NIDDK-funded autoimmune disease research areas. Clearly, words related to type 1 diabetes come up most frequently, followed by inflammatory bowel diseases. So I'm not going to have enough time to discuss all the extramurally funded autoimmune disease areas listed here. The rest of my talk will focus on selected research supported by DDN and DEN. Next slide. Although more than 80% of NIDDK's extramural budget goes to fund investigator-initiated work, mostly R01s, we do issue directed initiatives, and this slide describes the process. NIDDK's scientific staff identify research funding needs and concepts with input from scientific and lay communities through workshops and meetings like the Autoimmune Disease Coordinating Committee. NIDDK's leadership prioritizes these initiatives to align with the DK strategic plan, which is actually frequently updated and is currently being updated. NIDDK's initiatives are presented to Council, which is a diverse body whose members are drawn from the broad research community and the public. Once approved, funding opportunity announcements are issued, applications received, peer-reviewed, and awarded. Next slide. The next 3 slides, I will highlight research in DDN's division. I will focus on inflammatory bowel diseases and celiac disease for today's talk, but I noticed that some speakers today or tomorrow will touch on some of these other autoimmune diseases listed here. Next slide. To paraphrase Ramnik Xavier, a leader in IBD research, inflammatory bowel disease is a complex disease involving multiple genetic and environmental variables that perturb the immune microbiome axis. NIDDK-funded genome-wide association studies have identified over 240 genes that associate with IBD, which has created new opportunities to understand pathogenesis and explore therapies for these complex family of diseases. For example, in this paper cited down here, the identification of risk genes that increase expression of immune-activated integrin subunits predicts a possible clinical efficacy for integrin blockade in a subset of patients. Next slide. Another advance from DDN describes recent DK-funded work that used state-of-the-art single-cell analysis of inflamed intestinal biopsy tissue to identify a marker for the subset of patients who do not respond to standard anti-TNF therapy. Although the marker is complex, this approach could lead to better treatments involving combinations of therapeutic drugs to control different parts of the immune response in those resistant to anti-TNF alone. Next slide. Celiac disease is a serious autoimmune disease that occurs in genetically predisposed people where the ingestion of gluten leads to damage in the small intestine. One highlight from our portfolio in this disease is that a new animal model for which for the first time reproduces the gluten sensitivity of the disease phenotype. This model should help the field find ways to interrupt the disease pathogenesis instead of relying on symptom control through dietary restriction. Next slide. For the remainder of my talk, I will focus on research that's supported by a statutory funding stream known as the Special Diabetes Program. Congress has enacted laws that provide funding for 2 equal parts under this law. The SDP for Indians provides funds for diabetes treatment and prevention to the Indian Health Service, tribal and urban Indian health programs across the United States. The Special Diabetes Program also provides funds for special type 1 diabetes research through a series of laws that have provided approximately $3.39 billion over 26 years. Next slide. And next again, thank you. SDP funding supports ambitious, large-scale, high-risk, high-reward projects in type 1 diabetes. These projects are the type of projects that would not be otherwise possible. The funding enables stable, long-term investment in research, which is needed to identify the causes, generate new therapies, and to someday prevent symptoms entirely. I will be speaking about some of the SDP-funded programs that are listed here. Next slide. The process for the development of initiatives for SDP funds is similar to the regular process. It is collaborative and involves all interested NIH institutes and centers, as well as other agencies like the CDC. It also includes non-federal stakeholders. These stakeholders are engaged in part through a regular meeting of an external panel, which is coordinated by the DMICC, or the Diabetes Mellitus Interagency Coordinating Committee. So far, we've had 10 such meetings, approximately one every other year, and the next one will be held in April of next year. So these meetings allow us the opportunity to talk amongst ourselves here, NIH and federal agencies, and also with external stakeholders, and to try to get an idea of where the gaps and opportunities are for T1D research for the next bolus of funding. Next slide. Here are a few of the examples of research opportunities that received input and encouragement from the panel. We have released opportunities for collaborative clinical research with willing participants from some of our clinical networks, like type 1 diabetes trauma, which I'll be talking about more later. This we're terming the Living Biobank. It's been a great opportunity. We also recently released an initiative to fund research to improve the use of artificial pancreas systems in people with challenging to manage type 1 diabetes. We've also supported a consortium of researchers using biomimetic, or organs on a chip, systems to mimic islet cellular components and immune system interactions. Next slide. As shown on this pie chart, the majority of the SDP funds have been used to support clinical research, but preclinical research funding is not insignificant, and we have also supported resources such as islets for basic research and even career development. I'm also the program officer for career development for our division, and also standardization programs. Next slide. Here are some of the prominent examples of special diabetes program achievements in preclinical or translational research. The Human Pancreas Analysis Program obtains pancreata from cadaveric or organ donors to analyze with systematic and deep phenotyping, and makes the data that they derive from these samples and tissues available to other researchers. Unlike the Crohn's disease example I mentioned earlier, in type 1 diabetes, researchers do not have access to the disease tissue until it's too late. This program is relatively new, but it's already making progress, as shown here in the number of papers, and was recently leveraged to provide insights on the possible direct infection by SARS-CoV-2 of the pancreas to help explain why people with diabetes often encounter worse outcomes of COVID-19. Next slide. There have also been strong advances in genetics. The Type 1 Diabetes Genetics Consortium GWAS data, and this is publicly available through dbGaP, and follow-up genetic studies have accounted for more than 80% of the known genetic risk in the disease. This is pretty rare among polygenic diseases to have so much of the genetic risk already accounted for by known genes. Many T1D gene variants are actually shared with other autoimmune diseases such as rheumatoid arthritis. And recent efforts specifically seek to improve risk prediction using genetics by including underrepresented ancestries such as African ancestry, as shown here. Next slide. These two recent identifications of the function of causal variants identified in the genetic screening programs suggest pathways that align with autoimmune mechanisms and could lead to improvements for disease-modifying interventions. So I'm going to talk a lot more about disease-modifying interventions in type 1 diabetes. Next slide. Before I get to that, I'll talk more about achievements in diabetes control and complications. Look, the Clinical Trial Islet Transplantation Consortium validated islet transplantation processing and showed that islet transplantation dramatically improves hypoglycemia unawareness, which is one of the most dangerous and distressing symptoms for patients. And this was done with through the leadership of NIHs as well, our colleagues there. The SDP also supported the DRCRNet, which tested in clinical trials new treatments for diabetic macular edema and diabetic retinopathy, and that was with NEI. The SDP money is also used for small business innovation research. The SBI IR STTR program, nicknamed America's Seed Fund, provides money for startup companies, small companies, and has led to early development and testing of FDA-approved devices. Next slide. So these grants have vastly improved diabetes control and technology with numerous technology firsts, as shown here. Next slide. Another premier program funded by the SDP is very ambitious, and it's called the Environmental Determinants of Diabetes in the Young, or nicknamed TEDI. TEDI is a network of centers in the United States and Europe, which has screened infants for genetic risk. There's a lot of HLA alleles that are known to be very strongly associated with T1D risk. And so on that basis, infants were screened for this genetic risk, and hundreds of thousands of infants got screened, and the TEDI program enrolled 8,000. Now, these 8,000 participants have been followed from birth to age 15. There's 5 more years left, and all of the subjects will make it to age 15. And they have endured frequent sampling of blood, stool, other bodily secretions, medical records. They have a TEDI book in which parents write down every infection that they've had, other triggers, diet records, environment, stress, and more. The goal here is to try to identify the environmental trigger or triggers that spur the autoimmune process or progression to symptoms in children. So far, TEDI researchers have found interesting associations, including with a functional gut microbiome, with viruses, including enteroviruses and clinical infections, with the plasma metabolome, and vitamin D concentrations, amongst others. Notably, the refinement of a combined risk score based on TEDI data enhances T1D prediction and was one of the top papers of 2020 selected by the American Society for Human Genetics. TEDI has and is still generating lots of data, and this data is shared, and its analysis together with clinical data and other data will likely generate new hypotheses for triggers of diabetes for years to come. Next slide. For the remainder of my talk, I will highlight recent project from the Type 1 Diabetes TrialNet program. TrialNet was launched in 2003 following a 10-year diabetes prevention trial. TrialNet and other prospective study programs around the world have shown that T1D is a highly predictable disease. The course of disease is illustrated here by the TrialNet ribbon diagram, which is based on an original model presented by George Eisenbarth many years ago. The strategy that TrialNet takes is to screen relatives of people with type 1 diabetes, because family history is a proxy for genetics. If you have a relative with type 1 diabetes, your risk for diabetes is 15-fold greater than in the general population, where it's about 1 in 300. So far, TrialNet has screened over 200,000 relatives of people with T1D for the presence of autoantibodies. TrialNet tests for 4 different diabetes-specific autoantibodies, and we now know that once 2 or more different types of autoantibodies can be found in the blood, that individual is destined to progress to diabetes symptoms at some point in the future at nearly 100%. This is considered now stage 1 of type 1 diabetes. TrialNet then tests those who are multiple autoantibody positive for dysglycemia using an oral glucose tolerance test. And if that is detected, the progression to symptoms is much more imminent, and that's called stage 2 of type 1 diabetes. And once a patient is diagnosed, whether with symptoms or from testing, that's stage 3, or new onset diabetes. TrialNet has performed clinical trials of agents to stop the disease process at all 3 stages. And TrialNet typically starts an agent at stage 3 in the new onset to look at its ability to preserve endogenous insulin production. Preserved insulin production is associated with easier to control diabetes and reduce long-term complications. So there's definitely a clinical benefit there. But the real goal is to test the agent at the earlier stages of the disease when there's more beta cell function remaining, with the idea that that's going to help preserve function longer and prevent symptoms entirely. So I'm going to provide a few examples of clinical trials in the next slide. Next. Thank you. So these are some of the achievements in disease-modifying interventions in the new onset trials. As I mentioned, preclinical evidence in genetics supports a very strong role for autoimmune pathogenesis in type 1 diabetes, and this has been borne out in these successful clinical trials. This slide shows the mechanism of action of 5 agents that have so far shown efficacy in clinical trials to preserve C-peptide or insulin production in people newly diagnosed. 2 of these trials, as indicated, were led by the Immune Tolerance Network through NIAID. In each of these cases, the indicated drug was shown to preserve residual function even after symptoms appeared. And so far, as I mentioned, TrialNet's goal really is to take all these drugs and test them earlier in disease. And so far, one of them, next, yeah, this one, teplizumab, and then you can go on to the next, was shown to preserve residual function. Okay, so this trial then, teplizumab, in teplizumab, the strategy that TrialNet has adopted has really paid off. Treatment with teplizumab in stage 2 of the disease was able to delay the onset of clinical symptoms in treated participants by 2 years. So a median of 2 years delay. And a follow-up paper that was just published yesterday in Science Translational Medicine shows that this delay has now been extended to 3 years. A 3-year delay in symptoms and diabetes burden is highly significant clinically, especially for young children. The company Prevention Bio owns this drug and has received a breakthrough therapy designation, and we hope for FDA approval soon. And the leader of the trial was Kevin Harold, and the paper was in New England Journal, as shown here. Next slide. Okay, so back to the TrialNet ribbon. I mean, there's a lot more to do. Although this delay is significant and the preservation of remaining C-peptide is clinically beneficial, I mean, clearly we're not where we need to be in terms of preventing the disease. So TrialNet is ongoing. There's currently 3 ongoing trials, which are in green. The first 2 are abatacept and hydroxychloroquine. Those are ongoing in stage 1. And in those cases, we're really looking for progression to stage 2 as an early marker of disease progression. TrialNet has also launched a phase 1 stage 3 trial. It's shown in yellow here, but it's actually did enroll the first subject or participant in the study just a couple weeks ago. In addition, there's a combination therapy that's Ritux abatacept, 2 of those drugs that you saw that were effective in stage 3, now combined for efficacy, hopefully in stage 2, and a low-dose ATG trial that's in the planning stages. So these stage 2 trials have been delayed, unfortunately, due to clinical shutdowns because of the pandemic. As you probably appreciate, or maybe I didn't mention, but you know, the progression, it takes a lot of time. I mean, when we talk about progressing across these stages, you know, you're really talking about years, you know, 5 to 10 years in some cases from early stages. So the trials take a very long time, and this requires a huge investment as well to identify people who have risk, you know, have these autoantibodies. So we truly could not have made this progress, and TrialNet could not have been established without the Special Diabetes Program funding. In addition to that funding, I want to acknowledge the amazing participants and their families who participate in these very long-term trials, and the hard work from the scientists across the globe that have given us new insights into the pathogenesis, which has led to so many new life-changing disease-modifying therapeutics for autoimmune diseases. And now that includes type 1 diabetes. Next slide. Thank you very much for your attention. This is my last slide, and this is how you can get in touch with us, and I'll be happy to take questions.
Thank you so much, Dr. Spain. I think we'll hold questions till after the 3rd speaker,
Sure.
just to be sure we give each speaker enough time to speak, and then we'll have the opportunity to have the committee and even the audience submit some questions. So I'll ask Dr. Humble to begin his presentation. Dr. Michael Humble.
Okay, let's do a mic test. Can you hear me?
Yes.
Great, great, because I knew earlier today nothing was coming out. Well, good afternoon, everybody. My name is Mike Humble. I am a program administrator in the Division of Extramural Research and Training at the National Institute of Environmental Health Sciences. It's a pleasure to be here today to talk to you about our extramural portfolio in autoimmune diseases. Next slide, please. I know Lisa Ryder touched on this earlier, but I thought I would reiterate that the mission of the NIEHS is to discover how the environment affects people in order to promote healthier lives. Next slide, please. Our extramural division consists of 5 program branches where we are overseeing the science. Each branch has sort of its own general focus, although there is overlap. We have a scientific review branch that oversees and runs the study sections that are reviewing our NIEHS specific programs and our funding options. We have a grants management branch. They're the people who actually put the awards out. And we also have a program analysis branch. The autoimmune portfolio really resides primarily in 2 of these branches, the program branches. I'm in the Genes Environmental Health branch. Our branch is really looking at laboratory-based studies using model systems, animal models, things like that, to really tease out fundamental mechanisms, gene-environment interactions in terms of how environmental exposures lead to various health outcomes. We also have a portion of the autoimmune portfolio that falls within the population health branch. Those would be the population-based epidemiological types of studies. Together, all of our branches work together to really oversee our research and training efforts in environmental exposures and the various health outcomes that come from those exposures. Next slide, please. When we think about the environment, it means a lot of different things to a lot of different people, but I thought I'd just sort of help the group focus in on what we really focus on in terms of our extramural programs. So in a simple way, sort of think of sort of pollutants. So for example, we would be interested in pollutants in the water, things like lead or arsenic, for example, or air pollution, whether it's indoor air pollution from let's say cook stoves. That's probably more of an issue in our global environmental health program, but also air pollution. Maybe the elementary school is built next to a busy highway, for example. It might be pesticides on our food. It could be persistent organic pollutants that are in our environment, things like PCBs and dioxins. It's really any number of different types of chemical pollutants and toxicants that we're exposed to just as we go through our daily lives. It's not strictly, that's kind of a simplification. We also are involved and have an interest in climate change, for example. We've had a program on the built environment. Sometimes people see and wonder if infectious agents and pathogens, which we are certainly exposed to in our environment, fall within NIEHS's mission, but they do not. Those fall under the mission of NIAID and other institute missions. So that's sort of the type of research that we support, basically the things that we're exposed to just as we go through our daily lives because of where we live and where we work. Next slide, please. We know that we're exposed to things really throughout our lifetime, from in utero exposures through later in life. And so all of these different exposure time points are of interest to our extramural program. Next slide, please. As you might imagine, all of these exposures of interest can really impact so many different, probably all the different organs and systems in our body. And that leads to any number of different health outcomes. So this slide, I know it's a lot of little tiny words, don't be too bogged down in that. We have on our website what we call a Who We Fund page. And this page, these are screenshots from that page, sort of show the various portfolio emphases that we have. As you'll notice, there are a lot of different portfolios listed on this Who We Fund page. It's really because we have a very, very broad mission and very broad portfolio in terms of the different outcomes and different things that we focus on. The autoimmunity, it's sort of on the top right side of the column. There's the second one down. What it shows is that we have 13 active research grants that are focused on autoimmunity. Not necessarily the smallest portfolio that we have. You can see right underneath that the environmental impacts on bone and cartilage is much smaller. Likewise, we have some very, very large portfolios. Our neuro portfolio is very large. Our respiratory cardiovascular interests are much larger. But again, we have a very, very broad mission and a broad scope in terms of what we cover through our extramural division. Next slide, please. The autoimmune disease portfolio, as I mentioned, is split between myself and my colleague, Bonnie Jobert. I oversee the research efforts that are primarily lab-based animal model types of research efforts. Bonnie, as a trained epidemiologist, oversees our population-based epi studies. We work together really to understand what's going on in this field and future efforts and things like that. Next slide, please. If you actually were to click on that Who We Fund page, you would see a list of our current active research projects. I won't get into these too much, we just don't have the time for that, but I'll mention that the exposures would include mercury, crystalline silica, trichloroethylene, the solvent. I mentioned viruses earlier don't fall within our mission, but we have one project really looking at the co-exposures between crystalline silica and viruses and really genetic susceptibility, all of which are known to play a role in the development of autoimmunity. And so this project is really looking at the synergism between these multiple aspects and exposures and how that leads to an alteration of immune function. Targets are B cells, T cells, alveolar macrophage, looking at things like the aryl hydrocarbon receptor, estrogen receptor, and various pathways and really players in terms of what's going on in exposure-based changes in immune system function. Disease endpoints, I've listed some. Some projects are looking at systemic autoimmunity, other specific types of autoimmune outcomes. We also have some projects really looking at interventions. Are there dietary interventions that can really help ameliorate the impacts of these exposures and autoimmune outcomes? Most of the applications listed here are investigator initiated, although we do have a few that have come in from various funding opportunities that have been available to our research community. Next slide, please. When I looked at some of the other slide presentations from the extramural folks, I saw that there was an interest in terms of how our funding opportunities created and decisions made in terms of the directions that we should go. So I thought I would share with you what I have done with the autoimmune portfolio, where it started from and where it currently stands. So as a program administrator, we are responsible for the areas that we see and trying to really enhance what's going on. When I began overseeing this portfolio, it was very, very small, probably 4 or 5 grants, and I quickly realized that we should be doing and need to be doing more in this field. So 3 times a year, we have what we call a science planning meeting, where program administrators or maybe a representative from a working group will get up before our division and say, got this idea, I'd like to run a workshop or a symposium or create a funding opportunity to really enhance our research portfolio in specific areas. We get the feedback from all across NERT, our extramural leadership, our extramural division director, deputy director, and basically all the branch chiefs, listen to all these proposals and ultimately get back to us with their thoughts in terms of which ones will be going forward. My initial proposal was to do a workshop. I really wanted to figure out, before I started thinking about a specific funding opportunity for research grants, where did the field stand? So my first proposal was a workshop, which was approved, and I'll talk about that in a few slides. What I learned from the workshop was that there were a lot of areas that we could do more in. So it was a matter of thinking, okay, well, what can we do at this point? So I went back to our science planning committee with a proposal for a funding opportunity to really look at, to sort of stimulate new research grants, looking at environmental exposures and development of autoimmunity and autoimmune disease. I went into that with a budget in mind, with grant mechanisms in mind, and ultimately our extramural leadership got back to me and said, yes, seems like a reasonable thing to do, go ahead and go forward with the development of this funding opportunity. I will say there's some negotiation that goes on at this point. I had one budget in mind. Our leadership might have another budget in mind, but we work all those types of things out to figure out, okay, how do we want to move forward? Lisa Spain sort of implied or indicated in her presentation what happens next is that we go to our advisory council with a concept clearance, really telling our advisory council what types of things do we feel are important for our institute to begin doing research on or to focus our priorities on. We get their feedback, and ultimately they agreed that doing more in the role of environment and autoimmune disease is something we ought to be doing. So with their approval, I was able to go ahead and actually develop the RFA and get that out on the street. The actual timing that the RFAs go out on the street, as you can imagine, we have lots of programs queued up in development. It's really a matter of thinking, you know, big picture, where do things fit in the budget, spacing out the various funding opportunities, and really considering like our scientific review branch, because if we put an RFA out on the street, the demands on them are to actually create a study section to review those grants. We have to figure out where the funding opportunities will fit into the big scheme of things. The funding opportunity went out on the street, the applications came in and were reviewed, went back to our council to explain how things went and to sort of outline our funding plans. And at that point, the awards were finally issued. So it was a long process in this case. Not all the processes necessarily have to take that long. Some might take longer depending on the state of the science. We also use the same process if we were going to join on to a funding opportunity created by another institute, for example. If we're invited to participate in a trans NIEHS initiative, or trans NIH initiative rather, we would go to our science planning committee, tell them about this opportunity, the science that's going to be covered, what our interest would be, and really, again, talk with our leadership to say, I think we can, you know, fund a couple of grants that might come through this. Here's what the dollar figure might be, and ultimately get their approval to sign on to some of these trans NIH initiatives. So that's basically how we go about determining the areas that we're going to be emphasizing for our funding opportunities. Next slide, please. So as I mentioned, the first thing I did was we had a workshop. I call it an expert panel workshop to examine the role of the environment in the development of autoimmune disease. I really wanted to figure out, you know, what's the state of the science? What do we know about in terms of the role environmental exposures play in the development of autoimmunity? So this was a trans NIEHS effort. I know there were some questions earlier about how the different intramural and extramural programs work together. Fred Miller was a key member of my planning committee for this workshop. So we had intramural scientists, scientists from our division of the National Toxicology Program, as well as extramural scientists who were invited to take part in this workshop. We had epidemiologists, animal modelers, we had basic immunologists, mechanistic types of researchers, as well as some exposure assessment experts, really looking at, you know, what's the state of the science. And the goal was to say, well, based on what's been published, can we say with confidence at this point that these exposures are associated with autoimmune outcomes? Are there other exposures that, you know, based on the publication record appear likely to be associated with autoimmunity, but maybe require a few key experiments to really tease out, you know, what's really going on? And in thus doing that, I really try to identify gaps in areas that we could focus on for the future. Next slide, please. The workshop resulted in 6 different publications to really summarize our findings from that workshop. And I know several of the committee members were involved in this workshop. And so I have shared those with the group, and I won't go into those too much, but next slide, please. To highlight just sort of the types of things that came out of these workshops, the epidemiology paper would say, for example, that they were confident that crystalline silica exposure, for example, contributes to a number of different autoimmune outcomes. The solvent exposure looks pretty confident that's contributing to systemic scleroderma. In the animal models community, they're confident that the autoimmune responses we see in the animal models following environmental exposures are really influenced by the species and the strain of the animal in the study. I don't think that's a surprise really to anyone. It was acknowledged that forms of inorganic mercury induce systemic autoimmune disease in rats and mice. So these are some of the findings, examples of findings that the committees were confident of based on the literature. Other things seem likely, like the role solvents play in the potential development of MS. Again, likely, but some key experiments may be needed. Within the animal models, there's some indication that neonatal or early developmental exposures to dioxin, TCDD, can be promoting autoimmunity. This falls in line well with our interest of exposures across a lifetime, including early phase in utero types of exposures. So these are the types of things that came out of that workshop and enabled me to sort of get the handle on where things stood. Next slide, please. So this led to the development of our funding opportunity. There were a lot of things that needed to be done, but with the $2.4 million that I was given, I realized, well, with that, I can fund 10 exploratory R21 awards. So it's like, well, what do we want to do? So for this particular RFA, I focused on in vitro studies and animal models to really explore possible mechanisms in which the environmental exposures associated with human autoimmune endpoints, to study those types of things. Ultimately, if we can identify the mechanisms, the players involved, then we can, ultimately target things for maybe reducing those outcomes. One of the things, the caveats I did put on this funding opportunity is that the exposures to be explored needed to be associated with some sort of human autoimmune outcome. What became apparent from the workshop is that we had, on one hand, exposures being examined in animal models that were leading to autoimmunity. On the other hand, we had these exposures that were showing up in epidemiological studies, and the two didn't always match. So my thought was, well, rather than come up with new exposures in animal models, let's try to tease out the mechanisms for things that appear to be actually relevant to the human condition. So that was one of the caveats there. Now, next slide, please. We were able to fund 10 R21 exploratory projects from this RFA. The exposures and disease endpoints are listed there. In addition to these 10 applications, I was able to work with the PIs from the unfunded applications and really convince them that, you have an R21 already written. Let's look at the review statements, the comments that were made, and let's consider submitting that as an investigator-initiated application to the regular study sections. A number of the applicants did in fact do that, and so we were able to fund some additional R21 awards as well. Again, the idea with the R21s that these are exploratory. Ideally, if these projects work out and the PIs can generate some preliminary data, they can use that data to support more fully developed R01 type of awards. And a number of these projects have gone and actually done that. Next slide, please. So this sort of sums up the extramural activities in autoimmune disease over this since the beginning of the when the Autoimmune Disease Coordinating Committee was established. We have not put in out any additional funding opportunities. Again, at this point, our portfolio is really primarily investigator-initiated applications. There are some funding opportunities that my PIs have found that they apply for that really fit their ideas, but primarily investigator-initiated. We have another opportunity, I guess, listed in here where the intramural and extramural programs were working together. When our strategic plan was released in 2012, the emphasis on sort of a one NIEHS mindset was really emphasized. And so various faculties were created. And we had an information faculty. You might imagine these environmental exposures lead to a lot of information, which ultimately results in a lot of the health outcomes. And so the inflammation is kind of a broad-based, and we heard earlier today how important that is to autoimmunity. A lot of members, intramural and extramural, were involved in inflammation. We ended up forming a subcommittee on autoimmunity. This whole effort, Fred Miller was one of the chairs of this whole inflammation faculty. There really wasn't any money behind this to promote any science project, but we were able to, as a faculty, doing another workshop. Didn't focus on inflammation, but it was an opportunity for intramural and extramural to work together. Next slide, please. For my last slide, I thought I would just talk about some of the gaps and areas that I think we could go forward in. The previous slide highlighted a number of workshops that we've been a part of over the years. If you look at the meeting reports from all of those workshops, what you'll see is really sort of a common thread in terms of the recommendations. And primary is that we really need more epidemiological studies that will link specific exposures to autoimmunity and autoimmune diseases. As I mentioned, we also know that in life, it's not just single exposures we're exposed to. So mixtures, the exposome, multiple exposures are also of interest, although it makes the science very difficult to tease out what's going on. Lisa Ryder touched on the idea that we really need multi-site collaborations and standardized protocols for the collection of environmental exposure data in population-based registries in autoimmune disease. And I think that's something that would be a big help across the whole field, really trying to tease out what environmental exposures are really involved in autoimmunity. We need to continue to support basic and clinical research on the mechanistic relationships between environmental factors and autoimmunity. Again, teasing out the genetic polymorphisms that may lead to susceptibility to environmental exposures, creating better animals, really identifying the players in terms of the gene environment and health outcomes. One of the things I think it's important to do is to facilitate interaction between the variety of specialties. That was something that I heard after the workshop that we had from a number of people, that they really enjoyed talking and hearing the research being done outside their field. So instead of all the animal models people talking amongst themselves and the epidemiologists talking amongst themselves, we put them all in a room together. And so it wasn't just reading the paper, they actually got to talk about their interests and what they were doing. And I think they all really enjoyed that. And I think going forward, it's really important to break down some of those scientific silos and really learn from everybody to understand what's going on. And then finally, we are always interested in critical windows in terms of the timing of exposure and the health outcomes and the development of biomarkers to link environmental agents with autoimmune disease. So next slide, please. So with that, I will wrap up my presentation. These are, again, some of the URLs that were highlighted in the presentation. And next slide, please. With that, I thank you. And I guess at some point, I'll be happy to answer any questions that you have. So thank you.
We thank you, Dr. Humble. We will have an opportunity to answer questions right after the next speaker. There'll be plenty of time for questions from the committee for each of the speakers that were presented during the day. Next will be Dr. Rajapaksa, and she's the program director at the Transdisciplinary Collaborative Centers on Precision Medicine, the Centers of Excellence on Environmental Health Disparities, the Division of Scientific Programs at the NIH. Dr. Rajapaksa.
Good afternoon. Thank you, Dr. Rosal, for the kind introduction and the invitation to speak today. This has been it has been a fantastic session. In fact, again, I'm Nishradi Rajapaksa. I'm a program director in the Division of Scientific Programs at the National Institute on Minority Health and Health Disparities. A long time ago, I was a postdoc in Perry Blackshear's group in the NIHS Intramural program, where much of my work involved genetic studies on rheumatoid arthritis and sepsis. So it's great to see Dr. Fred Miller on the panel today, who was a collaborator of the Blackshear lab at the time. So for the next 20 minutes or so, I thought I'll share some information about NIMHD's efforts on health disparities research with a focus on autoimmune disease. So let me start with a few background slides describing the mission and the research focus of our institute. So the National Institute on Minority Health and Health Disparities is one of the 27 institutes and centers within the NIH. The organizational chart here depicts 24 institutes, the NIH Clinical Center, the Center for Information Technology, and the Center for Scientific Review. NIMHD became an institute in 2010. So as one of the disease agnostic institutes within the NIH, we support research in a variety of topics across many diseases and conditions. We have a relatively small budget compared to the other NIH institutes of about 300 million, which supports all of our research activities of the intramural and the extramural programs. Next slide. The mission of NIMHD is to lead scientific research to improve minority health and health disparities. We are currently focusing on 5 specific areas. NIMHD plans, reviews, coordinates, and evaluates all minority health and health disparities research and activities of the National Institutes of Health. Conducts and supports research on minority health and health disparities through intramural and extramural programs. Promotes and supports the training of the diverse research workforce, translates and disseminates research information, And then also a very important a part is also fostering collaborations and partnerships across the NIH and at the trans-federal level for different types of initiatives that otherwise NIMHD would not be able to carry out alone. Next slide. The NIMHD designated health disparity populations include Office of Management and Budget defined racial ethnic minorities. That includes African Americans, Hispanic, Latinos, Asian Americans, American Indians, Alaska Natives, Native Hawaiians, and other Pacific Islanders, as well as underserved rural populations, sexual gender minorities, and socioeconomically disadvantaged populations. So moving on to the next slide. So there are a few definitions for health disparity. NIMHD defines a health disparity as a health difference on the basis of one or more health outcomes that adversely affects disadvantaged populations. Often health disparities adversely affect groups of people who have systematically experienced great obstacles to health based on a variety of reasons, including but not limited to racial or ethnic group, religion, socioeconomic status, gender, age, mental health, physical disability, sexual orientation, or gender identity, or other characteristically historically linked racism, discrimination, or exclusion. Next slide. So in 2015, NIMHD began a visioning process to create a scientific vision that will transform minority health and health disparities research. The American Journal of Public Health, AJPH, has a special issue, New Perspectives to Advance Minority Health and Health Disparities Research. It's a series of essays and editorials that lays out a comprehensive and transparent agenda for the field of health disparities research. 30 specific research strategies across the 3 pillars guided the science visioning, and the 3 pillars include methods and measurements, etiology, interventions, in an effort to stimulate research for improving minority health and closing the gaps in health disparities. Next slide, please. So as a result of the visioning process, NIMHD currently supports 3-pronged approach, which include methods and measurements, etiology, and interventions in all of our funded research projects. First, the cause of health disparities are complex. Therefore, identifying best practices for methods and measurements would help researchers select the most appropriate methods for their research questions, and then to understand components that are critical to the scalability and also the successful implementation of evidence-based interventions. Understanding the etiology or the causes of health disparities is another component which is extremely important. So we are hopeful that large representative studies such as the million-person cohort plan for the NIH All of Us program that is prospectively collecting biological information, economic, behavioral, environmental, and other risk factors, as well as disease conditions longitudinally, would offer ways for researchers to identify causes of health disparities. Also, the All of Us program includes a very diverse sample. I believe it's about 50% diverse groups are involved in it. So then finally, the interventions also need to address structural. So when you're thinking about interventions, we need to think about structural factors such as poverty, racism, discrimination, residential segregation, unemployment, food deserts, and educational inequalities. So interventions also need to sort of consider mitigating some of the negative impacts of these factors, and then assess the mechanisms by which these factors may exacerbate health disparities. So some of the interventions research focuses also on identifying the best practices, scalability, dissemination and implementation science. Next slide, please. I won't go into this too much, but it's also well established that the social determinants of health plays an important role in the etiology of health disparities. So the Institute of Medicine's Committee on Recommended Social and Behavioral Domains and Measures for Electronic Health Records has identified selected domains and measures that capture the social determinants of health to inform meaningful use of the electronic health records. So to effectively reduce disparities and promote health equity in the nation, it's critical to incorporate standardized collections and the input of race, ethnicity, socioeconomic status, and other social determinants of health measures in all systems that collect health data. So in 2018, NIMHD Director, Dr. Eliseo Perestable, called on NIMHD to lead an effort to develop a consensus toolbox of measures on social determinants of health that would be endorsed and widely used by clinicians, NIH researchers, and that ideally would reside at the National Library of Medicine. So this was an effort primarily focused on data harmonization that would allow also for cross-study analysis domestically and internationally, accelerate translation research, and also would provide greater understanding of the causes of health disparities. So the SDOH measures collections on individual and structural measures were launched last year and are now housed in the Phoenix Toolkit. Next slide, please. One of the other things I wanted to also focus on is the NIMHD Minority Health and Health Disparities Research Framework, which reflects on an ever-evolving conceptualization of factors relevant to the understanding and promotion of minority health and the understanding and the reduction of health disparities. So this framework serves as a vehicle for encouraging NIMHD and NIH-supported research that addresses the complex and the multifaceted nature of minority health and health disparities, including research that spans different domains of influence. So on the y-axis, there are several domains of influence, such as the biological, behavioral, physical environment, socio-cultural environment, healthcare system, as well as different levels of influence depicted on the x-axis from individual to interpersonal, community, and population level. So this particular framework also emphasizes the importance and the need for attention to both biological and social determinants of health, rather than an exclusive focus on one or the other, which has been sort of much of the NIH research over the years. So currently, NIMHD funds basic translational epidemiology and clinical research. However, there seems to be a priority for social behavioral work. Next slide, please. So what type of research do we fund? Our institute is organized in around 3 research interest areas: the clinical and health services research, integrative biological and behavioral sciences, and community health and population sciences. So each of these divisions tend to fund research specific to its area, although there's quite a bit of overlap, as many of our funded projects have multi-level, multidisciplinary research activities. So often these research areas may occur in collaboration with other NIH institutes and centers. Next slide, please. So in 2016, NIMHD also launched a new program after President Obama announced the NIH Precision Medicine Initiative, the Transdisciplinary Collaborative Centers Program for Health Disparities Research, focused on precision medicine, is a collaborative effort supported by NIMHD, NHGRI, and NCI. that collaborates research on exploring precision medicine to promote health equity and advance the science of minority health and health disparities. So the TCC, supported through this initiative, focuses on several priority research areas, each combining expertise in precision medicine, population health disparities, and the science of translation, implementation, and dissemination to address one or more of the documented health disparities. I would like to highlight a project conducted by the Precision Medicine TCC at Stanford that has spearheaded a project to establish the first tribally owned biobank as a conduit to education on the benefits of precision medicine and big data health sciences. So which involved a pilot for the first set of biobank material through a collection of 12 additional participants for the sale or the studies of autoimmune illnesses in the Lakota Sioux community. So this biorepository for American Indian education, law, economics, and technology, known as the Bracelet project at Stanford, which was established in 2015, is a partnership with the Lakota community, Missouri Breaks Industrial Research Inc., and Black Hills Center for American Health and the Sphere Center at Stanford. So many of the other TCCs are also focused on other aspects, from pharmacogenomics to genomics and electronic health records, and then efforts in the Caribbean. So in 2020, we published a collection of articles in a special issue in the Ethnicity and Disease supplement, highlighting some of the scientific advances from this initiative. All right, so let me highlight a few studies that has been funded by NIMHD with its long-standing interest in supporting research on autoimmune conditions such as osteoarthritis and lupus. It's clear that many of the autoimmune disorders disproportionately affect racial ethnic minorities and low-income populations, and it continues to be an issue, as often the case that it's understudied, underdiagnosed, misdiagnosed in these populations. So NIMHD portfolio on autoimmune disease is quite small, however, offers an opportunity for us to collaborate with other NIH institutes and more resourced institutes such as NIAID and NIDDK. Also, it's known that a lot is unknown in this space where it comes to racial ethnic minorities, and there's an urgent need to fill the gap of knowledge in understanding how multiple exposures or influences of social, environmental, biological factors intersect, contributing to etiology and downstream pathways of autoimmune disease. Next slide, please. Just briefly, in 2011, the first pilot initiative between EPA and NIMHD was successfully launched that supported a variety of health disparities research efforts to increase multidisciplinary research that Dr. Humble a few minutes ago alluded to, exploring environmental, biological, behavior, and social factors. So subsequently, we supported 5 additional EHD centers in 2015 in collaboration with NIHS and EPA, and then 3 more in 2020. So we had several goals in mind for this particular initiative: to stimulate basic and applied research of the multiple factors underlying environmental health disparities, to promote innovative approaches to mitigate environmentally driven health disparities, increase community engagement, mentoring, and capacity building. Also, the study of environmental health disparities by examining the results of the synergistic effect of exposure to multiple environmental and social hazards in the context of poverty, psychosocial stress, and discrimination, and the built environment. So next slide, please. So one of the centers that was funded through this program is the EHD Center at University of New Mexico, led by Drs. Johnny Lewis and Melissa Gonzalez. It's one of the EHD centers conducting studies in New Mexico area of the Navajo Nation. So just to orient you, half of the native population in the US currently lives in 13 Western US states, where 161,000 abandoned rock mines are located, hard rock mines are located. Approximately 600,000 Native Americans live within 10 kilometers of these abandoned mines. So one of the studies, the DNA study that was done on the eastern part of New Mexico, of the Navajo Nation, many of the tribal communities in these areas are impacted by comorbidities as a result of the toxic environment exposures as well as social impacts. And then the tribal communities rely on local resources for food and water, creating additional concern over potentially hazardous exposures through the contaminated livestock and water, resulting in even further impacts. So some of the ongoing environmental legacy exposure studies have shown that it leads to increased risk for autoimmunity, multiple chronic disease, and hypertension. And in one of the studies with around 1,300 individuals, they have reported autoimmune disease associated with a proximity to the abandoned uranium mines. And then a more recent study from Erdai et al. Examined specific autoantibody measures to ascertain exposure associations with uranium and arsenic contamination in the drinking water, which later proved to be an additional added risk to the location proximity to the mines and waste site. So overall increase in ANAs captured more than 220 nuclear and cellular antigen responses. So these are all strong predictors of the environmental exposures that needs to be teased out in the future. So these autoimmunity measures are very important in order to monitor and use them to find at-risk individuals as well as groups potentially progressing towards autoimmune disease development in the future. So there was some discussion earlier after Dr. Ryder and Dr. Miller discussed the need for autoimmune registry, which I also think is quite important and critical for future studies in this area. Next slide, please. So to highlight another study, so lupus is, as everyone knows, most often affects women. It is more common among African-American, Latino, Asian, American Indian, Alaska Native women than among white women. So one of the projects that NIMHD funds is an online training about lupus for promotores, lay health educators who work in the Latino community. So through the promotores being trained to do outreach to low-income Latinas to inform them about lupus, provide support to those who have the disease, and then help those who have lupus to navigate the health system and teach them also how to advocate for themselves. So this is it's a 6-module bilingual course that has proven to be rather effective. Next slide, please. And NIMHD is also supporting research and development of mobile health and other technologies to reduce the burden of musculoskeletal disorders among socioeconomically disadvantaged and geographically underserved populations. One such project involves developing a self-applied wearable ultrasound therapy device for osteoarthritis management in this underserved population. So this project has proven to be a non-drug, long-duration ultrasound device for the treatment of chronic osteoarthritis pain. And the technology has been preliminarily tested with positive results. And then last year, this device was FDA approved, and which offers promise in effectiveness in both efficacy and cost. Next slide, please. NIMHD also has funded a few genetic studies over the years to understand genetic variation between ethnicities that could account for the underlying differences in disease severity and clinical manifestations. for a variety of autoimmune diseases. So we know that African American and Latino patients are significantly more likely to progress to end-stage renal disease than white patients with glomerular disease such as lupus nephritis. So one of the studies by Dr. Ali Garavi, so they looked at the different immune-mediated glomerular disorders that represent the third most common cause and end-stage renal disease, accounting for 14% of the dialysis population. So the IgA nephropathy, membrane nephropathy, and lupus nephritis. So these are the leading causes of primary and secondary immune complex glomerular diseases. And there are significant ethnic and racial differences and prevalence in the outcomes of these diseases. And so several of the SNPs, or the single nucleotide polymorphisms, that have been discovered from this group has really advanced the science in this area. Thanks. I think that's my last slide, and I'm happy to take any questions. I spoke really fast.
Thank you very much. This was really a very informative and timely discussion, so much appreciated. Let's open up to the question and answers period at this moment. And if committee members have questions to start off the discussion, once again, just raise your hands. And if you identify a specific speaker that you'd like to address the question to, please do so. And at the same time, identify yourself for the remainder of the question and answer period. So let's open up, and we'll look to see who has questions for any specific. Glinda, let's start with you, and then I have Deidre and Michael.
You're on mute, Glinda.
Okay, I'm Glinda Cooper. I just have a quick follow-up, I hope, for Dr. O'Shea and Dr. Barron. You had a nice discussion of your intramural program and all the advances and particular investigators. Do you have epidemiologists as part of your intramural program?
Carol, you want to go first?
Sure. Yes, we have epidemiologists. We also have two tenure-track epidemiologists that just started. We also have other epidemiologists that act for more service for our investigators, but we do have two tenure tracks that just started.
And what are their focuses or foci? What division are they in? Just what are their interests?
They're more interested in infectious diseases at this point.
Okay.
Yeah.
Okay. So
but one of them is pretty broad. One of them is very broad, but mostly infectious diseases.
Okay. And Dr.
We have, we have an individual who works on outcomes research, and he worked both in conjunction with our orthopedic surgeon and separately. He just retired, and so we're thinking about where we're going to go in the future. We may sort of pivot towards more emphasis on health disparities, working on that. As I was looking through my slides, just making sure that I had actually covered the things I wanted to cover. I skipped one slide at the end. Speaking of epidemiology, I'd like to make the pitch. I think the whole issue of autoimmunity is going to have stating the obvious, a huge impact by our pandemic. So whatever rate of autoimmunity we have now, I would suspect that's going to change a lot in a lot of ways. Just saying.
Okay. And I also, oh, go ahead. Well, go ahead.
Linda, you have one more question? Go right ahead.
For Dr. Humble, I was wondering, are you planning another workshop? If, now or a few years from now, to follow up on that 2010 one, and particularly, bringing in what's been learned from these, the collaboration with NIAM, the Minority Health Program, the Environmental Health centers on health disparities or environment disparities. I think it'd be fascinating. So are you planning on?
Well, I think that's a real opportunity now to think about what would our next workshop be. And yes, I think discussions like this are a terrific way to begin launching and thinking about what would that next workshop look like. So it's always a possibility and of interest in terms of, what would we go to next as a workshop. But yeah, I think that's a possibility.
Thank you. Deirdre?
Great. Thank you all first for your presentations. I had a couple of questions for Dr. Rajapakse, and then actually on the second question, others I'm hoping may be able to chime in as well. But I guess my first question was on one of your early slides, you mentioned that NIMHD plans, reviews, coordinates, and evaluates all the disparities work at the NIH. I was wondering if you could sort of talk about how that is operationalized.
Right. So it's actually it's in our mission to do so, but although I think in actuality it has been rather difficult, but with now under Dr. Perestable's leadership, there's much more aggressive, not aggressive, but there's much more interest in collaborating and trying to ensure that these discussions are started across the NIH. And because I think oftentimes, I think as somebody mentioned earlier, our institutes tend to work in silos, and oftentimes, the discussions and the communications haven't been up to par. So, but I think moving forward, there's much more interest in that particular area, and that we are hoping to take a much more leadership role in that space.
Thank you. Yeah, it was the evaluate part that I was particularly curious about. So if that, and whether that's a part of your charge as an institute to sort of evaluate the portfolio more broadly.
Because currently many of the research portfolios are evaluated by its specific IC, and then also there are certainly challenges to evaluation as well. So which study should go as under racial, ethnic minorities, health disparities versus under a particular disease. So there are certainly analytical challenges associated with that as well. But yes.
Okay. Thanks. And then my second question, and this one, maybe I'd be curious about from the other people from the other institutes could comment on this, but you mentioned that within the NIMHD portfolio that there's work around translation and dissemination of, presumably thinking about if, for example, a clinical trial or something was conducted, that a part of what falls under your portfolio is support for dissemination of that. Did I interpret that right? And then I guess I don't hear that often from institutes. So I was wondering if others have that as a part of what they are supporting for the extramural researchers. Did I hear that right?
I can start, but I think, so many of our studies, definitely, this is a strong community engagement aspect to it. And then we are also trying to promote health, not just healthcare, and then also to try to sort of understand the importance of health and not just healthcare as well. So a lot of the findings from our research, we try to make an active effort in disseminating some of the information, especially like through the TCCs on precision medicine. I know that Consuelo Wilkins' group at Vanderbilt Meharry, they have done a tremendous job in that space.
And this is Mike Humble. I'll just say quickly about NIEHS. We may not really necessarily have many clinical trials. We tend to be more on the front end of the disease process. But community engagement, community dissemination has always been a big part of what we do, particularly with like our center programs, where we've had actually community outreach and engagement cores, specifically to really get the message out that as we start identifying exposures that may be impacting specific communities, really getting the word out to them in whatever type of appropriate manner there might be, to alert them to what's going on in their neighborhood and in their area.
And also from NIDDK side, I don't know how relevant it is for autoimmune diseases necessarily, except that, of course, for T1D, a big part of that disease is treatment and self-care. And so we definitely fund a lot of research on the best way to get the technology to the communities that need it. And then also we're investing in stakeholder engagement, which is before we start a clinical trial, to make sure we're in the community, understanding what patients want and what they need. And they're informing not just dissemination, but the trial, from the very beginning.
Thank you. Lisa.
Thank you so much. These were fabulous presentations. And Dr. Rajapakse, again, I'm going to go to you. I found your presentation about NIMHD to be really, really interesting. And I wondered whether you could reflect on something that I suspect might be a little controversial. And that is that disability is not considered a disparity condition for NIMHD. Although a lot of your program announcements say that disability can be a factor that researchers could look at. And I just wondered whether you might be able to comment on that and whether there might be some thinking about changing that, because there is really good evidence about people with disability experiencing discrimination, structural discrimination, inability to even access healthcare facilities because of inaccessible equipment, et cetera. And the Healthy People Initiative since 2000 have considered people with disability a disparities population. Obviously, disability is really important for people with autoimmune disorders. So I just wondered whether you might be able to comment a little bit about that for your institute.
Right. No. So I know that there has been a number of discussions on this topic in the past within the HHS. And then, of course, many of the designations are made at the OMB level. And so I think more recently, actually, so even SGM populations were added as a disparity group. So we are hopeful that, perhaps the discussion around disability would also happen at some point. So it's not a lack of interest on our part, but I think the designations itself generally take, it's a complicated process. So, so currently, I think, as you said, where we stand is that, we fund research in that area, but not particularly designated as a disparity group. So we like to see other,
Yeah, like you've got a program announcement out there right now that mentions disability, but it's not considered by your Institute of Formal Disparities population.
Because it's not really institute that made the designation, so it's the OMB categories, and so it has to come from downtown.
That's really helpful. Thank you very much. Thank you.
Yeah.
Michael?
I have a kind of philosophical question. First, I wanted to say I'm actually thrilled to hear how well so many things are going. But I'd like you, and I am glad that so many of you addressed both gaps and opportunities. If you had no political restrictions and administrative restrictions and all the money, what could NIH do that would really change things? And I'd like to put a little asterisk to that, because I like John O'Shea's statement, is autoimmunity a useful concept anymore? I'm literally thinking of big ideas that would change the way in which this whole area is approached, and anyone can ask.
Well, you mentioned me, so I'll speak up first, and then I'll let Dan correct me if I'm making mistakes. I think the whole idea of inflammation, just like we have a moonshot on cancer, a moonshot on inflammation would be, I think, important. And actually, as I was saying before, it's tremendously timely in thinking about COVID, in the sense that, science, the advance in sciences, have made it possible that we're probably going to survive this pandemic, but there's going to be lots of consequences of this. So I actually think it's tremendously timely to be thinking broadly about what happens when people's immune systems are disrupted by the pandemic, and then to be thinking broadly about how does inflammation affect all these diseases? What's the integration between inflammation and metabolism, coronary artery disease, et cetera. And Dan, you think about this way more than I do, I think. But so, correct
Yeah,
me.
well, John, I certainly agree with that, and already, at least to some extent, some of the pharmaceutical companies have picked up on this in terms of, for example, looking at the role of IL-1 inhibitors in cardiovascular disease and diabetes, and I think that is a real opportunity just broadly, and then as you say, John, focused on the pandemic is certainly a timely opportunity. So, in terms of having something that would be really a transformative idea, I like that. I think that's really good.
That's great. Thank you.
So I'd like to comment. So we are immersed in COVID in my institute. Almost everybody is touching it somehow in their research program. And the idea of branching out and broadening the concept of autoimmunity to inflammation is a fabulous idea. But when you think about some of the diseases and some of the things this virus is causing that we didn't really think are going to be happening, like MIS-C. MIS-C is a child immune system overreacting to something that we don't know. I mean, to me, that's autoimmunity in itself. And we need a lot of research to try to define what's causing that. And then the question is that same thing going to happen when kids get vaccines? I mean, everybody in our institute is like, what's going to happen next? What's the next experiment going to show? So I think there's a lot of excitement in this area, but there's only so much COVID money, and we have to decide where exactly it goes first.
From the public, there was a question related to the availability of PowerPoints and some audio of the conference today. If it is available, that is one question, and if so, how to access?
Yes, so I did actually put a response in the Q&A for everyone to see, but the slides and the video will be posted to the NASEM committee study page, and the link is in the response that I provided.
Okay. Any other questions from the committee members?
Scott has his hand up.
Excuse me?
Scott.
I have a question, if that's okay. Scott Lieberman, University of Iowa. It's been a very enlightening day, and I appreciate everyone's presentations. A theme that seems to have kept coming up repeatedly is the successes that have been had with interinstitutional or multidisciplinary collaborations, and another term that has come up repeatedly is people working in silos. And I'm curious if anyone has thoughts and recommendations for us on how we can sort of turn those silos into multidisciplinary interactions, and what can we do to make those successes kind of more common?
Well, I guess
go ahead, Dan, go ahead.
I guess that with any of these kinds of things, carrots are better than sticks. And so creating incentives for people to work together, whether that's, some sort of glue grants or, something like that, that would encourage collaboration between institutes, or the creation of resources that only can be accessed if people are coming from different disciplines or institutes. I think those are the kinds of things that make sense to me anyway. Certainly the model of having workshops that we heard about, just getting people to talk to each other is also a very powerful way of catalyzing these kinds of things.
What I was going to add was I think AMP is an interesting experiment and something that it was not without some road bumps. And again, based on my own experience, if you're really going to if you really want a drug, you have to collaborate with industry. And so now, of course, that brings up issues of conflict of interest and all this sort of stuff. I think the mechanism at the NIH, the CRADA mechanism that I mentioned, I thought was very effective. And so, yeah, you're absolutely right. You know, you don't make these big advances without serious collaboration, and that serious collaboration doesn't just mean academics, it means industry, but it goes both ways, because it's very clear to me at this point that despite the advantages we have with industry, they're not going to go back and ask some of the most interesting questions. So it sort of has to be this two-way street.
I thought, this is Mike Humble from NIEHS Extramural. I thought I would kind of respond to this in terms of the carrot versus the stick idea. So within our extramural program, we created a program that we called our VICTOR program. I had to pull it up real quick on the internet to make sure I remember what it stood for, but it stands for Virtual Consortium for Translational or Transdisciplinary Environmental Research. I combine all those letters in VICTOR. But what it was an opportunity for like our R01 grantees to add additional specific aims to an already funded project. But the idea was to bring in new collaborators on that project, people who you hadn't collaborated with before, to bring new science to that original parent project. But it was just that kind of thing, and an attempt on our part to really start to foster branching out and bringing in new expertise into a research project. That's still an ongoing opportunity for our researchers as well. We've changed from a competitive revision now to a straight R01, but the idea is new collaborations.
Emily?
Hi, Emily Summers at the University of Michigan. This might be a question for Dr. Spain, because I believe you mentioned being a program director for career development, but more broadly for anyone. I'm just wondering if you might have ideas or thoughts on career development for extramural faculty. There's the aspect within the NIH, it's clear that you are investing in the individuals and that helps with longevity over time. And I'm wondering about, in particular, like the mid-career range, which I feel kind of might be prone to falling through the gaps. And one example I can think of is that K-24s, I believe, are geared more towards clinical faculty, and there are not opportunities that I'm aware of for PhD trained faculty to lead those sorts of mid-career grants. So I'm just wondering if you have any thoughts on just the mid-career stage. We talk a lot about early career stages elsewhere, and how you might build in longevity for investigators at later stages where they can also cultivate their more early career mentees.
Yeah, that's a really, really good question. I mean, the NIDDK has had a K-24 program, as you said, for clinical investigators for quite some time. And but we took a pause and we're reevaluating right now. I mean, we're really looking at this to see what is the best way to really support these mid-career investigators. So I hear you, and I think this is something that hopefully, we're going to take a strong look at. I'm looking at programs too to think about mentoring just broadly. Who's I think great scientists, we talked about collaboration, but they also, it's like a sort of a lifelong mentoring really has to happen. And there's not a lot of support for that, or sharing of even best practices for mentoring. And this is particularly important for women and underrepresented groups, coming through the scientific pipeline. And I think, especially for also building the entire pipeline, I think junior investigators need to see that there's a career path, that it's not like, oh gosh, like there's a lot of stuff early on and then you just fall off the cliff. So I think, we have to do it in an integrated way. That's my opinion. But I'm interested in hearing from some of my other colleagues if they have ideas about what they're doing in that space. Thanks for the question.
So I don't hear Dan speaking up, but I would like to, Dan came up with a great idea for his scholars program when he was clinical director in NIAMS. I mean, because of exactly that concern of that, we really do need physicians who are, understand basic science, and that just plain takes time. Again, Dan, it was your program. You want to talk about it?
Sure. So this is perhaps a little bit earlier than the question, but the idea is that we had in NIAMS a number of individuals that had finished their rheumatology fellowship, at least the clinical part of their rheumatology fellowship, who weren't really ready for careers as independent investigators. And so the idea was, depending on whether they were interested in a laboratory-based career or a clinically based career, that we had two different pathways for them with protected time, with mentoring, and with an increased salary, sort of intermediate between a fellow's salary and the salary of someone that would be a junior faculty member, to bridge that gap for individuals that we felt were promising candidates in an academic type setting. I think that the question, though, was for people that were more mid-career that might be a little bit further along in their careers. Is that right, or am I misunderstanding that?
Yeah, that's one of the things I'm thinking about. And again, the example of the K-24 program not being available to non-clinicians, I think can really impair the ability to have some of these more interdisciplinary awards that might help support the faculty themselves, but also to mentor the next generation. And we've talked a lot about, and I'm an epidemiologist, so I'm biased towards cohorts and long-term observational studies. But I think it's a big deal when you have perhaps to follow through some of these longer-term cohorts. It takes way more than a 5-year cycle. So outside of R01 mechanisms, what can we do to be supporting extramural researchers, in particular to allow them the opportunity to invest in some of these bigger undertakings that really require a lot of infrastructure to be built?
Yeah, so in a way, I mean, what you're talking about is the kind of commitment that I was talking about at the beginning of my talk about the intramural program and being focused more on the investigator than on specific projects to allow for a longer term follow-up. And I think that kind of model, at least to some extent, probably would be useful in the extramural world, and how exactly to replicate it, I think that probably depends on the specific field as to what would be the best way of doing it. But I think there are great advantages to it, that's for sure.
This is Mike Humble, and I'll just follow up and say, there's been so much emphasis on early stage investigators and special programs for people just getting started, whether it's career development awards or special R01s to really help launch those careers. But then what happens when that R01 is time for renewal, or exactly what you're pointing out, what's happening to those mid-career people who are still really trying to get their research programs going? And I don't have an answer, but just acknowledge the fact that, this is becoming a greater interest across the NIH in terms of those mid-career scientists, scientists at risk, who are kind of at the early phase, is still really trying to get the real lock on the longevity of their research programs. So the discussions are beginning, and you bring some very, very interesting points up. I just wanted to acknowledge that.
Yeah, there's been a lot of discussion and a lot of data produced with regard to that early stage valley of death, or whatever you want to call it. And it may be that actually for someone to look and study and see, how many people are dropping out mid-career, it would identify a real problem that needs to be addressed.
Well, once again, I really want to thank our speakers and all who tuned in today. This has been an extremely informative session for the committee. And as you know, it informs us considerably as we go forward with our work, specifically in the writings we have to do over the next number of months. We may reach out to some of you for some additional information. So if you see a name that looks familiar from today, you'll know it came as a result of your presentations. In terms of the public, I hope it was as informative to those of you who attended as well. And by the way, if you have any comments or questions, those can be sent directly to Awo, who is the project study director. And once again, the email address is autoimmunestudy@nas.edu. autoimmunestudy@nas.edu. Please keep in mind, related to that, that any material you wish to share with the committee will be made public through the project public access file. That is because the National Academies complies with Section 15 of the Federal Advisory Committee Act rules. So just keep that in mind if there are any materials you wish to share. Once again, thanks all so very much. It's been a pleasant and informative day. Look forward to speaking to all of you once again in the near future. Enjoy the day.
Thank you very much.
Thank
Stay
you.
safe.
Nice seeing everybody.
Thank you.
Thank you.
Thank you.
Bye.
Bye-bye.
Bye.